Disease Day, we are reminded that there are 7,000 rare diseases impacting more than 300 million people around the world. Since 2008, this internationally observed day has been shining a light on rare diseases, advocating for life saving research, and working to advance health equity. In Cleveland, we are fortunate to have participation from the Cleveland Clinic, university hospitals, and Metro Health, along with Case Western Reserve University. Thank you for bringing together brilliant clinicians and researchers to raise awareness of rare diseases. And thank you to everyone involved with the National Organization for Rare Disorders and the Samaria Foundation for your ongoing activism and advocacy for care, research, and cures. The work that you are doing truly makes a difference around the world and right here in Cleveland. We know that healthcare costs for those with rare diseases are often 3 to 5 times greater than for those without such conditions. Getting a correct diagnosis can take years and lots of visits to different doctors. Finding treatment can be challenging, and if treatment options do exist, they often involve costly travel and hospital stays. This is a burden that is simply too much for many families already struggling to make ends meet. And while the individual diseases may be rare, their circumstances are not. The NIH estimates 10% of Americans are impacted by rare disease. That's between 25 and 30 million people. The work that you do gives these families much needed hope and resources to care for their loved ones. Thank you for all you do and for raising awareness of rare diseases in the city of Cleveland. Take care and God bless. On rare
Hello, I'm Ohio Governor Mike DeWine. Every year on the last day of February, we recognize Rare Disease Day. There are thousands of rare conditions that you've probably never heard of, but there might be one Ohio family that's severely affected by one of those rare conditions. In fact, one Ohioan has a rare condition that affects only 2 other people in the whole world. The struggle these Ohioans and their loved ones go through is often very, very difficult. Through Rare Disease Advisory Council, we are providing resources and hopefully improving lives. I'm grateful to the dedicated doctors, nurses, researchers, patient advocates, nonprofit groups, and others that make up a community of Ohioans who work tirelessly for each and every patient and every family. I'm thankful Ohio is developing first-class therapies and treatments, including work at Nationwide Children's Hospital with SMA. Other great work is being done at Cincinnati Children's Hospital, Ohio State University Wexner Medical Center, University Hospitals, the Cleveland Clinic, Akron Children's, and so many other hospitals across Ohio. Thank you for learning from each other, supporting your fellow Ohioans, and most of all, for your commitment to the rare disease community.
Hello, my name is Samira, and I am a patient living with seronegative neuromyelitis optica spectrum disorder, also known as NMOSD. I was diagnosed in 2014, and just 2 months after my diagnosis, I launched the Samira Foundation, which is now a global nonprofit organization dedicated to raising awareness of both NMOSD and MOGAD. We're very passionate about building communities of support for patients and their caregivers because I don't believe that people should go through rare disease alone. We fund research and, of course, are involved in patient advocacy. Happy Rare Disease Day 2023. I'm actually recording this video from Rome in Italy, where I've been for the last couple of weeks, traveling around the country, meeting with doctors, patients, caregivers, researchers, advocates, to learn about the rare disease landscape here and see how we can help Italians in the context of NMOSD and MOGAD. I have been celebrating Rare Disease Day since 2015, and I have to say it has been an absolute pleasure and brings me great joy to have observed over the last few years how much Rare Disease Day has grown in terms of impact, in terms of reach, in terms of engagement. You know, every year Rare Disease Day gets bigger and bigger and bigger. And that makes sense. You know, apparently there are about 10,000 known rare diseases with 600 million folks around the world affected by rare disease. Those numbers are not small. Those are very big numbers. And while I wish rare disease didn't exist because I don't like the idea of folks suffering, I am comforted by the thought that we're all in this together, and there is a huge strength in numbers. Even in our own NMOSD and MOGAD communities, I've seen so many leaps and bounds happen just by the virtue of working together as a community. So, you know, watching the rare disease community do as much as they do is really inspiring and very impressive. I am so proud to be part of this community. I am the first to tell anyone and everyone, at any chance I get, that I am a patient living with a rare disease. It's like a badge of honor for me, and I hope it is for you too. So I just wanted to come on here and say thank you for loving rare disease and caring about it so much. It's because of this passion and commitment to rare disease that we're able to accomplish as much as we can. And here's to a wonderful, successful, impactful, and exciting Rare Disease Day 2023. Thank you for all of the work that you're doing and for inspiring me and the Sumaira Foundation team every single day. Lots of love, strength, and hugs to you all.
Good morning, everyone. Let me first thank you all for coming, visiting the Case Western Reserve University campus. We are delighted to be the host this year for Rare Disease Day. We're going to start the morning off talking about one particular type of rare disease, and that is genetic diseases as well. And we thought it's a really timely topic because of the advances in technologies now that are making it appear as that we're going to make a lot of headway in a number of these diseases in the coming years. Maybe I better start first by also thanking NORD, the National Organization for Rare Diseases, and in particular Pam Judge and the Judge family for putting this whole event on. If you haven't seen, Pam is a force of nature, and she makes a lot of things happen. It's not a trivial thing to get a video from the governor and from the mayor. That's quite a testament to the drive this woman has. So let me start with try to put things in perspective to you. So for the non-biologists in the crowd, you may not realize it, but your bodies are made up of about 30 trillion, maybe upwards to 100 trillion cells. And each of those carry all the same genetic information, right? So you have 3 billion bits of information in every one of those 30 trillion cells. So as Sumaira just told you, the numbers are huge for these types of things. And across your chromosomes, all that composes about 20,000 different genes that are the instructions for you to be who you are. And if even one of those little bits of 3 billion pieces of information gets messed up, it can cause pretty catastrophic consequences. So, but those numbers, if maybe you're more aware with probability now that sports betting is legal in Ohio, but the odds of these things happening are quite rare. But when you put them all together, there are so many of these rare diseases that affect our kids. And some of them you've probably heard of, but most of them you haven't. So things like sickle cell anemia, Tay-Sachs disease, cystic fibrosis, phenylketonuria, just a couple of the 7,000, 8,000, 9,000 different genetic disorders out there. And as you heard, these are affecting thousands and actually millions of our kids in our population. And we want, obviously, to do something about that. And that's been the mission of NORD all along since it was formed. Well, we have put together a program that's bringing together faculty from Case Western, from University Hospitals, Cleveland Clinic, Metro, to try to work together. You've got a lot of really smart people in this town. If we can put those brains together and work together, we can make a lot of headway, we think. Right? So how are we going to tackle this? What are we doing? Right now, we treat diseases like this from the end of the symptoms, right? So we know they're caused by changes in your DNA. Well, the DNA makes a copy of RNA. That RNA then gets read by the cell to make the protein. The protein is the business end of things, but that's what then causes the symptoms of the disease. But until recently, we've really been restricted to working on the symptoms. But now we have some new technologies available to us that are allowing us to think about actually repairing the DNA. Well, how are we going to do that? And I'll talk a little bit about that. But administratively and functionally, how we're going to do it, it's by bringing all these people together. In 2015, we created a thing we call the Research Institute for Children's Health to pattern our therapeutic development process on some really substantive successes we made for one genetic disease called cystic fibrosis. And with cystic fibrosis, we've taken the picture from not knowing what caused it to finding the gene, understanding what that gene does, understanding what the protein does, and now we have some really, really impressive drugs that are quite effective for many of the individuals affected with CF. So the changes in DNA that cause genetic diseases aren't specific to cystic fibrosis. So we ought to be able to apply those same methods to all these other diseases as well. So now, where are we? Now we can think about this. You've heard about some of the technologies, CRISPR-Cas, things like that. So now we can think about targeting the DNA, the RNA. If many of you have experienced the Moderna or the Pfizer vaccines, that's an mRNA therapy. So we know those things can work and they're safe. We now, cystic fibrosis and some other diseases have shown us that we can actually find drugs for some of these diseases that will affect the protein and fix it. So we can actually get at the cause and not the symptoms of many of these disorders, right? So right here, we're working diligently on this technique that folks have read about called CRISPR-Cas. And this is a technique where we can go in and try to take advantage of the cell's natural properties to fix DNA and see if we can harness that to repair a mutation that causes the disease. So we like to portray the process as a little pair of scissors in the cell. Doesn't quite look like that, but functionally, this is a way for us to decide where we're going to break DNA, and then we can trick the cell when it's repairing that break. We can trick it to fixing the sequence in there and hopefully circumvent the mutation that causes disease. Okay, and there's plenty of examples where these types of therapies are now working. So Dr. Anna Mitchell will tell you in a little bit about a variety of different strategies that are being used for this. And we're now at a point then where it's not can we do it, it's how can we do it well enough. That the efficiencies of these things, that's what we're struggling with. So again, remember, you got 30 trillion cells, and if I'm going to try to do this in 30 trillion cells, I'm up against quite an obstacle. So can I focus on just the cells that are affected in a particular disease? If I'm working on a metabolic disease, maybe I only need to work on the liver, for example. Well, Dr. Laura Konzel is going to tell you about some gene therapy trials that are going on here right in Cleveland through at UH for a disorder called ornithine transcarbamylase deficiency or OTC deficiency, right? And that's a great place to start. You're working on almost exclusively one organ system to be able to make it more tractable. But if you're working on disorders like cystic fibrosis, where you have to get into the lungs, into the intestines, you have a different set of difficulties to deal with. If you're working on a neurological disorder, got to get in the brain, into the nervous system, right? And what if you can't do it well enough the first time? What about having to repeat the process a number of times, right? So those are the kinds of hurdles, the engineering things that we're trying to figure out, right? And then lastly, what about, how do you know you fixed it, right? What are the processes? And for a lot of disorders, the long-term effects, you need to wait weeks, days, months, even years sometimes to see the efficacy. So we're trying to work with folks in our radiology group and things like that, where we can look at some of the molecular and physiologic signatures that are more easily or readily discerned. And I'm showing here one example. I went down to talk to a friend in radiology, a guy named Chris Flask, and was explaining to Chris the difficulties we're having with CF, that if we want to look and see whether or not the lungs are getting cleared out more quickly and stuff, sort of the standard was to do x-rays or things called CTs. And Chris says, well, I think we can do that by MRI. And for most of us, an MRI, you go in, your knee's injured or whatever, you go in and you're in that tube and you're there for 30 minutes and you hear the thunk, 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 thunk. I said, well, Chris, that's not going to work. We're talking about little kids, 2, 3 years old, that we can put in there. And that's a tough thing to do. And Chris gets on his keyboard and he reprograms the scanner and he said, okay, well, I can get you a scan that'll give you most of the information in about 10 seconds. And I think even the most active child, you might be able to get still for 10 seconds. But just showing you here on this that the set of lungs here to the left are what healthy, normally perfused lungs look like. But then as you go to the right, those are what an MRI image looks like as the lung disease gets more severe in these kids, right? So now MRI, you're exposing a kid to a magnet, not to radiation and stuff. So you can think about doing sequential measurements and often do measurements so you can see whether or not these therapies are working. So this is just one example, but those are the types of engineering things that we're doing around here. And I want to impress on that is one of the powers of an academic institution, that we have so many different expertises on which to bear. And so we like doing that ourselves, but it also affords opportunities to interact and cooperate with some of the biotech and the pharmaceutical industries to really make these things move forward into the patients. So that's all I'm going to say just as the introduction. So now you're going to hear first from Becky Dara. Becky's going to give you a talk on some of the fundamental concepts of genetics. She is, in my opinion, one of the best at making useful analogies that can help you understand what we're trying to do and how a gene and leads to a protein and how they cause disease. Then Anna Mitchell will talk to you about different successes we've had with genetic diseases. And then finally, Laura Conzel is going to talk about studies going on right here at UH and Case Western for some genetic disorders. So with that, Becky, I'll hand over to you.
All right, I can barely see over this podium. I need like a phone book back here, but I don't, half of you don't even know what a phone book is. Is that right? Okay. All right, so thank you for entertaining my talk this morning and being here bright and early. We're going to take a step back and think about what are some of the basic concepts of genetics, and hopefully that will help frame the rest of the conversation today. And maybe it'll give you a new way to think about these concepts a little bit. So And I promise the cheesy title will make sense in a minute. Stick with me on that one. So for many of you, for many people with rare diseases and their families, that I'm guessing their first encounter with genetics was under the guise of understanding inheritance. And for a long time, that was really where the conversation ended. When we thought about rare diseases, we would, as genetic counselors, we would think about what is the recurrence risk? What's the chance for somebody else in the family to have a child with something similar? What's the chance for this family to have a second child or a third child with something similar? And we talked about autosomal recessive or dominant or X-linked, and we talked a lot about those kinds of inheritance patterns. But after that, the conversation about genetics tended to stop. Maybe at some point there was genetic testing, and we could look at the DNA itself and identify what might be the change that caused that disease in that individual. But beyond knowing that actual change, there wasn't a whole lot we could do with that information. And it's exciting that that's changing, and that's what we're going to hear about today. The idea that understanding what that genetic etiology is does have implications for developing therapies and developing treatments. And so we'll move beyond sort of this general discussion of inheritance and talk about why we might need to understand the genetics at a different level. So to do that, here's one of the reasons why we need to do that. What we're going to talk about today are these kinds of genetic treatments, and there have been just amazing advances. I work primarily in the field of cystic fibrosis, but in many, many other disorders that we'll hear about today as well. The advances in using this kind of genetic approach to treatment, it has been amazing. Dr. Drum already mentioned the COVID vaccines, but that's probably the reason we're all in this room being able to meet together in person is the fact that those became available and were as effective as they were. And that was a genetic-based therapy that was introducing mRNA into the system in order to confer protection against COVID. So this is some examples of some of the headlines from this kind of approach. So as a genetic counselor, and there are a number of genetic counseling students in the room today, raise your hands, genetic counseling students. So if any of you want to discuss genetic counseling with any of our trainees today, I'm sure they'd be delighted to talk to you. But as genetic counselors, one of our jobs, and I would say probably the most important aspect of our job, is being able to take things that are fairly complicated and explain them in a way that somebody else could understand. Maybe somebody who doesn't have a degree in genetics, or somebody who's trying to understand the nuances as they may pertain to their family or their child or themselves. So we're going to do a little analogy today, and this is where the cheese comes in. So I know it's a little early for a cheesesteak. Maybe we should have been making a Danish or something a little more breakfast friendly. But we're going to think about a gene as really a recipe. And have any of you looked up a recipe online lately? Have you? All right. What do you get? You don't get a nice recipe. You get a whole long story, right, these days? And it's got all these descriptors of, the way your grandmother's kitchen smelled when she made it, and the marketplace where you might shop to find it, and hints on what the best produce might be for this recipe. Just let me find the recipe. I just want to know, what I'm supposed to be shopping for, or what I'm supposed to be doing. If you read through, you can find oftentimes the actual ingredients, but it takes a lot of work, all right? It takes a lot of work to read through. If we think about this as the gene or the DNA sequence is really like that whole big recipe on the left-hand side here, the ingredients list is similar to the RNA. And then the thing that you make, this good-looking cheesesteak down here in the corner, is the protein. So we can think of it in those terms, that this recipe, this DNA, has a whole lot of filler in there that it's interesting. Maybe it helps me shop for the best ingredients. It might have some purpose, but it's not part of the direct ingredients list. All right? The direct ingredients list is really exactly what I need to make that recipe. And then the recipe itself, the cheesesteak, the thing I can eat, the protein that functions in my body is the protein. So we can use that analogy then to understand how different types of mutations may impact this system. All right. So there's a type of mutation called a nonsense mutation, and that's one that you'll hear about, I think some today, but that's in the literature a lot because this is a very challenging kind of mutation. A nonsense mutation is essentially as though there was a period in the middle of that ingredients list and then no one shops for anything beyond that. So you can imagine this would be quite devastating to making our cheesesteak if all we have is 2 hoagie rolls sliced and 2 tablespoons of Lord knows what. So this does not make a hoagie. This is going to be pretty upsetting. There's no cheesesteak there. There's just 2 buns. So there's nothing there I can use afterwards. That period ends the ingredients list, and I can't move beyond that. There's a different kinds of mutations called splice mutations that we'll hear about today as well. Splicing is that process of taking out the filler in between all of the ingredients on the DNA list and making the list of ingredients that represents the RNA up there on the right. If I have a change in that splicing, it means I will have either extra things in that ingredients list that don't make any sense. So in this case, I've got Whiz, I suggest, partially frozen, at a standalone location, try hunting, be made in Philadelphia, and ask your butcher to slice you. None of those are all words that are in, those are all phrases that are in this recipe, but they're not going to be useful in my ingredients list. I do not want to ask the butcher to slice me. So I don't want those as part of my cheesesteak. I want those taken out. If I have a splicing mutation, they get included in, and the cell can't make sense of that. Or sometimes with splicing changes, things are missing from my ingredients list. In this case, I've got hoagie rolls and butter and American cheese, and it actually does make something decent, right? I've got a grilled cheese. Maybe I even prefer that at times. But it's not the cheesesteak we thought we were going to make. So sometimes these changes are worse than others. Sometimes they confer something that's actually useful, and may, in a certain cases, be beneficial. If it's Lent and I don't want a chili feast steak, listen to me, Philly cheesesteak, I might want a grilled cheese instead. But it's not what I intended to make from that specific recipe. Finally, there can be a deletion of genetic material, and we'll talk about that as well. And certain deletions are more impactful than others. Certain ones can mean more. For example, if I delete the word green from my ingredients list, it doesn't really matter what kind of bell peppers I use for this recipe as long as I'm using bell peppers. But if I delete the word pepper, all of a sudden I've got a green bell in the middle of my recipe. And that's not going to be useful. It doesn't make sense. It doesn't belong in my cheesesteak. That's not going to work. So you can see that different types of mutations are going to confer a different problem with the protein. And each one is going to require a different strategy for correction. If we go back to the main recipe, we can talk about correcting some of those spelling changes. We can talk about putting back in some of those deleted sequences. We can talk about removing that period that causes the nonsense mutation in the ingredients list. We can talk about changing up splicing to remove those parts of the ingredients list that aren't supposed to be there or correct them. And these are all real possibilities now. I don't have to just look at those changes and wonder, I know what their impact is, but there's nothing I can do. I can now tackle this at the level of the recipe itself and come up with strategies for correction. So some of those that we're going to talk about today in terms of strategies that would impact the DNA, there's gene delivery. Gene delivery is essentially bringing an entirely new recipe card to the show, all right? So we're going to show up with a whole new recipe, the whole thing with all the filler, everything in there, so a whole new gene delivery. Gene editing would be like spell check or some sort of way to read through that recipe and make sure that all the components are there and there aren't any skipped words or misspelled words. And if there are, going through and fixing them with certain molecular strategies. There are strategies that work on the ingredients list itself. So rather than fixing the recipe, we can go right to editing the ingredients list or the RNA. And there are compounds called nonsense read-through, and all that means is there are ways that I can sort of white out that period and unmask the rest of the ingredients list that might be behind it, all right? I can read through that period and see what else is supposed to be in that ingredients list. combinations called short nucleotides or mRNA strategies. Short nucleotides are things that protect that splicing. They kind of hide that chunk that we don't want in there so that my ingredients list stays pristine. It stays clear and only has the ingredients that I want in there. Those short nucleotides will cover that up so we don't have those splicing errors. And then mRNA replacement, such as the COVID vaccine, is really just bringing, rather than a whole new recipe, just bringing the ingredients list. So now we're going right for the bang for your buck. We're not going to worry about bringing the entire recipe itself with all the filler. We're going to show up to that cell with the ingredients list and say, please make this. All right. And as I mentioned, that one is working. So that was a huge breakthrough. I think the scientists were hopeful for decades. Those kinds of therapies were in development for decades, and people were hopeful that would work. And it was just very exciting that it did, that you could show up with an ingredients list and have something made. And then finally, there are a number of therapies that are already available that work on that protein itself. In cystic fibrosis, we have a number that are called CFTR modulators, but there are other, this might be your enzyme replacement therapies or something where you're tackling the protein itself and returning, you're showing up with your cheesesteak, if you will. So it doesn't matter that you didn't make it yourself. It doesn't matter that you didn't have the ingredients list. You're coming with it from someplace else. All right. And we're going to hear about all of those approaches today. And I will like to introduce Dr. Anna Mitchell. She's the clinical director for the Center for Human Genetics at University Hospitals Cleveland Medical Center. And she's going to talk a little bit more in detail, I think with fewer cheesesteaks involved, maybe no cheesesteaks involved, in the actual scientific approaches behind these. But hopefully that analogy helps you understand a little bit about what we're talking about when we think in terms of these approaches. All right, thank you.
Okay, good morning, everybody. Thank you for being here. Now, we'll talk about some of the options for treating genetic diseases. In terms of how you can potentially treat diseases, it's going to depend on what the problem is or what's wrong in the cell. And so just some of the examples, maybe no proteins made, the no cheesesteaks there, maybe not enough proteins being made, or maybe the protein's made, but it doesn't work right. And so depending on what's gone wrong will determine what your options are for potential therapies and treatments. And so just thinking about the flow of information that you've heard about, from the DNA to the RNA to the protein, those all three places represent opportunities for treatment in terms of intervening at the level of any of these, the DNA, the RNA, or the protein. So we're going to go over some of the potential examples of treatments at all of these different levels. So the first thing covers the opportunity to put back what's missing. And as Becky mentioned, enzyme replacement therapy. So if the protein isn't there, there's no cheesesteak. If you can make the protein in the lab, or a company can make it in a form that can be delivered to the patient, it then becomes an option to directly replace that missing protein or enzyme. And currently, this is most often used in the world of metabolism or diseases that affect how the body digests proteins or carbohydrates or fats. And so typically you have the enzyme is the fancy term for a protein that helps with digesting molecules in the body. And so that enzyme acts on a substrate or something, it digests it and breaks it down into these little blue and green products there that the body can utilize. So that top row shows what should normally happen. And then if you have an error in that enzyme, it doesn't work properly, then you can see on the bottom what happens is you don't get that flow of processing that you should, and you don't make the blue and green products in the end, and that yellow starting material accumulates. And either one of those can be problematic to the body. Having too much of the yellow stuff can be bad, or not having the blue and green. So either one of those can be damaging to the cells. But if you can somehow make the enzyme and replace it and give it to the body and get it in the right cells where it needs to work, you can fix that flow of the molecules, and you can then have the blue and green product that you need, and you can get rid of the yellow that's building up. And here are just some of the genetic conditions where enzyme replacement therapy exists. So companies have managed to make the enzyme in the lab in a form that can be delivered to the patients, typically intravenously. Oftentimes they need to go every couple of weeks, every month, and they get an intravenous infusion of this missing enzyme, and it can distribute through the body to the cells where it's needed, and it works until 2 to 4 weeks later when they go back and get another dose. And so this is very effective for conditions where we can make the missing protein, we can make it in a form that works, and we can get it into the body in the places it needs to act. And so that's one example. A second example of a way we can treat disease is to help a broken protein. So supposing the body's making the protein, but it's not working properly. And Becky alluded to cystic fibrosis as a condition. So the protein in cystic fibrosis is a channel, or like a doorway in a cell, and its job is to open and close to let things in and out as appropriate. And with some patients who have cystic fibrosis, that door gets stuck closed. So figure 1 on the left is showing how that door can open and close with the swinging ball on the bottom. And then on the right-hand side, panel 2 shows what happens if that door gets stuck and it doesn't open and close. Well, molecules can't go in and out of the cell as they should, and that can be problematic. But if you can design a drug or a compound that will help that door open up when it needs to be open, that becomes an opportunity of an effective therapy. And so in this case, the left is showing that door being stuck shut, and the little blue molecules are being bounced back out. They can't go through the doorway. But look on the right, when we treat with the compound that then opens up that doorway, now the little blue chloride molecules can go in and out as they need to. So this becomes a very effective treatment for individuals who have a problem where their doorway or their channel's stuck shut. This medication works. But if you are a patient who is not making any of the channel at all, or there are no doorways, this therapy is not going to be effective. So this works great for people who have the type of problem of a doorway being stuck shut, but it's not going to work for all patients. And so that becomes one of the limitations of this therapy, is it works for only a certain subset of individuals with cystic fibrosis. I'm backing up a step to the level of the RNA. So this is our ingredient list from the cheesesteak analogy. On the top there, panel A shows that RNA, normally the ingredients get assembled and you make a normal protein. So on the right there, you can see the blue curlicue protein that should be made that's your cheesesteak, if you assemble all your ingredients. And then in panel B, you see what happens if you have that period or that stop in the middle of your ingredient list. You make a shortened protein, or you make part of a cheesesteak, and may not be enough. That may not work. But what if you could have a compound that would allow the body to ignore that period in the middle and keep right on shopping and get all those ingredients? That becomes an effective treatment for this type of gene change or this type of problem. And so in panel C there, it shows what happens if you treated with a compound that now that period, that stop sign in the middle gets ignored, the body reads all the way through the ingredient list, assembles everything, and now you're back to having your normal protein or your full-size cheesesteak. And this is a therapy that's used for Duchenne muscular dystrophy, which is a disorder that affects the muscle. Individuals have weak muscles. They eventually lose the ability to walk. It can lead to respiratory lung problems as well from weak chest wall problems. And so this therapy becomes very effective for individuals who have this type of problem with their Duchenne muscular dystrophy gene. Again, this is effective. If you've got that early stop in the middle of your ingredient list, this is a good therapy. But if your gene is broken in a different way, if it's another problem, this medication is not going to be effective. So again, we can treat diseases depending on what the treatments differ depending on what the problems are, how the gene is broken in the first place. And then backing up one more stage to the level of the DNA, what are some options we have there? So we can talk about fixing the gene. And this here is an example from a disease called sickle cell anemia. And so that's a problem with the red blood cells, where you can see healthy red blood cells should be round, kind of like inner tubes. And you can see with sickle cells, they have gotten collapsed and they're shaped like a sickle. And that becomes problematic in tiny blood vessels. Those sickled cells get stuck, and then blood can't flow past them. And then the tissues beyond them become damaged. And so sickle cell anemia results from one single letter change in the whole sickle cell gene. And you should have a letter A, and individuals with sickle cell have a T. So one single letter change. And all individuals who have sickle cell anemia have the same DNA change. So if you can design a treatment for sickle cell to change that one letter that's broken, it's effective for all patients who have sickle cell anemia, because as far as we know, they all have the same DNA change. And that's not true for all genetic conditions. But if you are using that CRISPR-Cas technique that Mitch mentioned initially, if you can take the red cells from a patient, they're very early stage or precursor red cells, if you can take them out and you can use CRISPR-Cas to switch that broken T to the A, which that's the correct letter that you want in the gene, if you can make that change in the lab and then you can give those cells back to the patient, you have now corrected the sickle cell anemia. You've corrected the genetic defect. And this is very nice because you're taking the patient's own cells and you're fixing them and giving them back. And so that helps one of the problems we have with genetic therapies is the immune system is very smart, and if it sees something that it doesn't recognize, it tries to destroy it. And so that's a challenge in treating genetic diseases, is evading the human immune system. But in this case, you're taking the patient's own cells and you're treating them and then giving them back to them. And so that bypasses the whole immune problem, because the body's going to see that just as the cells that it's known all along. And so this is currently an option for treating sickle cell anemia, again, where we're editing the gene. And it works well because it's a primary blood cell problem, so you can treat just that one tissue type, put it back, and it works. So again, an advantage here that's not true for disorders that affect multiple different organ systems, and it's potentially harder to get your fixed gene in and throughout the body where you need it. And then a final DNA option is the ability to put the whole good copy of a gene back, or that whole full-length recipe that Becky talked about, using gene therapy. And that's the idea where we have that whole full DNA written recipe, we package it in a little something we call a vector, and we give it to the patient and have it get into the cells. And so showing there that new genes, the little yellow thing, typically the vector we use is a virus, because viruses are very good at getting into human cells. That's what they've evolved to do. So we take a known virus, but we take out the genes that make humans sick out of the virus, so giving it to somebody is not going to make them ill. And you replace it with the gene that you want to deliver. And then if you give the patient this modified virus, it's going to get into the cell, because again, viruses are very good at that. And if it transports itself through the cell and it gets to the nucleus, which is where genes are read and made into the RNA, then that new gene will be incorporated and the body will find the ingredient list out of it, and it will then use it to make that cheesesteak or what you were missing. And there's a couple of options you can, on the left, where you give the virus directly to the patient, and this you might want to do in a case where you need the gene to function in multiple different places. There's also the option of, again, similar to the sickle cell treatment, where you take cells out of the body, you infect them with the virus, and then you give them back to the patient. And either of these are possibilities. And currently, gene therapy is approved for a genetic condition called Leber congenital amaurosis, and that is a disease of the retina, the back of the eye, that leads to visual impairment starting very early in childhood and progresses throughout life. This is one of the advantages of this is you inject the gene therapy right directly into the eyeball. You can get it right into the retina. And again, this works very well because the main problem, the gene, where it matters in the body that the gene is broken, is in the eye. You can deliver the therapy right to the body organ that's affected. And so this is the one currently approved gene therapy condition in the United States. There are clinical trials underway for other conditions to try to expand this treatment for other disorders. And so with that, I will introduce Dr. Laura Konzel, who is one of our biochemical geneticists here at Case and University Hospitals. And she is an outstanding clinician who treats individuals with metabolic diseases, but she's also involved in many clinical trials exploring new options for treatment of genetic diseases, including some with gene therapy.
Hi, everyone. Thank you for being here this morning. I'm really excited to tell you about the clinical trials that we have available at university hospitals, especially since gene therapy is something that we're actually offering here in Cleveland for certain disorders. And it's a really exciting time to be a metabolic physician because this is an area where the development of gene therapy allows us to replace enzymes that are not working in the body. So I am going to just start to give you an idea of the clinical trials that we're involved with just in our Center for Human Genetics. There's many other clinical trials going on in other departments, but in our department, we focus really on rare diseases. And I wanted to point out the registries and consortia first, because these are registries that are involving multiple different centers around the world. And really, those are very important to be involved in when you have a rare disease, because this is how we learn. When you have a rare disease, a lot of times doctors at a certain place may have 1 or 2 patients with a rare disease. But when you work together with groups from all over the world, other locations within the US, and we pull our information together, that allows us to find patterns and develop new treatments for these rare conditions. And alone, individually, we couldn't do that without working together. So one of the largest groups that we've been involved with for the last 20 years, and of course, I've been part of it for a while, but it was really started by a group of doctors, including Dr. Kerr, who is a physician here, a metabolic physician here at UH, and a couple of other physicians at Children's National decided that we really needed to look at urea cycle disorders and put our information together to try to see how we can develop new treatments for these conditions. And having those registries has led to having new treatments. And currently, we're involved in a number of clinical trials where we have new diseases that have developed from these registries and the information that came from them. And highlighted in red for the investigational new drug clinical trials that we have here, those are the therapies that involve gene therapy that we have available here at UH. The first chunk in red are specifically for a condition called OTC deficiency, or ornithine transcarbamylase deficiency. I'll talk a little bit about that because I'm sure that's not something that's familiar to everyone in this room. It's a common metabolic disorder in the group of patients that I see, but generally most people have not heard of this condition. And then in the last part, there's another gene therapy trial that's coming here in the future. We're selected as a site, and we're not up and running just yet, but it's gene therapy for something called PKU, or phenylketonuria, which is probably the most common inborn error metabolism that many of you may have heard of before. So just to start off, I wanted to talk about the ornithine transcarbamylase, or OTC, gene therapy trial. And this was the first gene therapy trial that we were involved in our genetics center here at UH. And it was a phase 1/2 trial, meaning that we found that this drug was safe, and we also found what the most effective dose was. So there were 3 different groups. And basically, we did this by giving a normal working copy of the human OTC gene through a virus called adeno-associated virus type 8. So it's AAV8 is the abbreviation. It targets the liver, and it delivers that working gene to the liver to be able to make normal OTC enzyme. And I will go into a little more detail about that, but I wanted to first talk a little bit about what OTC deficiency actually is. So ornithine transcarbamylase is the long name of the enzyme. Most people just call it OTC deficiency. It's just easier to remember and say. But it's the most common urea cycle metabolic disorder. And what the urea cycle does is it actually removes ammonia that accumulates in the body. And ammonia comes from just the normal process of breaking down protein that we have in our body. So we all take protein in when we eat. Some foods are higher in protein than others. Our body makes protein. And if you cannot effectively break down the byproduct of ammonia, it accumulates. And if it gets really high and accumulates a lot, ammonia actually can damage your brain. And if it gets really high, it can affect your ability to breathe. It can put you in a coma. Someone who has a really high ammonia may end up intubated on a ventilator. And if you don't know that the ammonia is high and you don't treat it, people can die from this. And we have different degrees of severity of this condition. It's an X-linked condition, which means that this gene is located on the X chromosome. So for babies who are boys, and they only have one copy of the X chromosome, they tend to be much more severe. They get sick right away, just in the first few days of life. And their ammonia levels, normal ammonia is less than 45 generally. These babies have ammonia levels that go up over 1,000. And when it gets that high, they are in a coma, they're not responsive. Usually it happens after they start eating. Initially in the first few days, they're taking in protein and they just cannot process it. And that ammonia accumulates. And babies that are that severe, usually the way to treat this, the urea cycle lives in the liver. So you can give this baby a brand new liver. That is a cure, but it's a big deal. And a liver transplant is usually the option that we go for these really severe babies, but there's not always a liver, and it's a big deal to go through a liver transplant. So there are people who have milder forms of this disorder. So oftentimes the moms of these little baby boys actually do have OTC deficiency, and sometimes they have milder symptoms, and looking back, they had the flu and maybe ended up in the hospital. They were really out of it, but no one really checked in ammonia. They got some fluids with sugar in it. They got better and nobody really thought anything too much of it. Or they have their baby, and within a few days of having their baby, they end up in a coma. And when you have a baby, your body starts to reabsorb all this protein that was present in the placenta and the uterus. The blood volume increases when you're pregnant. All of that reabsorbs right after birth. So sometimes the first time you see a woman have a symptom is right after they have a baby, and they end up in a coma and their ammonia is up in the 700s. So it's a really serious disorder. You can die in that situation too if there's no intervention. And it's kind of common for a genetic disorder. It's 1 in 14,000 people, and it's probably underdiagnosed. So it's probably more common than this. And in Ohio, this condition is not on newborn screening. There's work to get it on newborn screening so that we can detect these kids and people, especially the people who aren't going to present so dramatically in the newborn period, but still have symptoms. Because if you have these episodic elevations of your ammonia when you're sick, or when you have a high protein load, that can damage your brain over time. Even these little elevations over 100 over and over again can cause damage. And so having the knowledge that somebody has this condition is really important so that we can treat them. Generally, we look at the OTC gene, and about 80% of the time, we can find a change in that gene that caused it. Sometimes, though, 20% of the time we don't find a change. And it doesn't mean somebody doesn't have OTC deficiency. We usually have I only listed these metabolites because they tell us that somebody has OTC deficiency if their ammonia is very, very high, they have orotic acid, and glutamine's high in amino acids and citrulline's low. So you can still have it and not have a mutation. We think there are mutations in regulatory parts of the genome where you turn on and off this gene. So you can have OTC deficiency even if you don't necessarily find the change in the gene specifically. And I won't go through the whole biochemical pathway, but I just wanted to point out where the OTC enzyme lives. And this is within the liver, and the whole urea cycle occurs within the liver. And OTC is the second step in the urea cycle. And usually patients who have a defect in how the urea cycle works right at the very beginning have the most severe symptoms, and OTC deficiency is one of them. So generally, when we have someone who has a high ammonia level and we need to treat them and they come in, their ammonia is several hundred, we oftentimes put them in the ICU for treatment. And the treatment is pretty involved. We have really expensive medications that work, that can be given IV to remove those ammonia molecules that are accumulating and causing the toxicity. And Ammonul is the specific medication. If the ammonia is really, really high, that may not do it quick enough, and we have to put them on dialysis. Oftentimes they're intubated in a coma, and we can give alternate forms of energy that the patient can use without having to use protein. So we give sugar through an IV, and we can give fat through an IV. And once the patients are under control and we know that they have OTC deficiency, from a day-to-day basis, it is a big deal. It's a daily change in how their life is lived. They cannot eat as much protein. So things like meat, these patients usually aren't really able to eat. If they eat a lot of protein, they start to feel a little confused. They might feel nauseated and throw up. They have headaches. So they're really not able to eat that much protein. And oftentimes we give them specialized formulas that have more carbs and more fat, and they have just essential amino acids that make less ammonia. And those formulas don't taste very good. So it's something, especially for someone who's diagnosed later, it's hard to comply. And then we have daily medications to remove ammonia. Ravicti is over $100,000 a year. So these are really necessary but very expensive medications that we use. And then we also give citrulline as a supplementation. But now that we have the availability of gene therapy as part of clinical trials, that may change for people who have OTC deficiency. The kids that are presenting as newborns, they still are going to need liver transplants. But for the moms who show up later on in their 20s, 30s, or people who are a little less severe, who maybe didn't have their first episode until they were a teenager and had the flu, gene therapy actually might be a viable option for them. And the goal of the OTC gene therapy is to provide that working OTC information as a gene that is carried to the liver by that virus. And once the virus gets there, it starts to live in the liver. And the way I explain it to my patients is basically we're giving you a one-time dose through an IV of a virus. So we're giving you an infection, but that infection will go directly to your liver, and it's a safe virus. So the AAV8 virus was selected because we get exposed to that virus. You can in the community. It doesn't make you sick. So you can be exposed, never know it. It doesn't really cause any symptoms. But it does go and live in your liver. So it's kind of like when people would get the chicken pox when we were kids. You get exposed, you get sick, but that virus stays with you and it lives within your body. And it makes, at a low level, makes proteins and enzymes throughout your life. But it doesn't really cause you a problem, except unless is reactivated and you get shingles. But in this case, because the AAV8 virus doesn't make you sick to begin with, it's a very safe virus to use to carry this working gene to the liver. And as it's living there, the goal, and we think as we're following these patients, what happens is it continues to make working OTC enzyme from these correct instructions, so this normal OTC gene. And as it does that, it reduces the ammonia load within the body. And it can get these patients off of those medications, the Ravicti, the Bufanol, that they don't have to take those anymore, or if they do, it's a much lower dose than they needed before. It can liberalize their diet where they can even eat a normal diet and previously could only have just a few grams of protein a day. In some patients, it's allowed them to have more than half of the protein that they used to be on. Some of the patients who had more severe symptoms to begin with maybe didn't get to go to a totally normal diet, but they can eat a lot more than they used to. And then the risk of ending up in the hospital from a hyperammonemic crisis is much lower. And the way that this medication is given is just through an IV dose one time. So in the phase 1/2 trial where we found the right dose and found that it was safe, there were 9 sites worldwide, and we ended up treating 11 patients. And one of those patients was here in Cleveland. And the sites that are in red are the sites that actually dosed at least one patient. And at this point, the phase 1/2 trial is closed, and we're compiling the data and reporting it. But there were 4 cohorts in this study. So the first 3 cohorts were just escalating the dose to see which gave you the best effect. We found that the highest dose actually gave the best effect, but also there was one little side effect that we saw happening in most patients. And when you're giving such a large dose of virus, even though the AAV8 virus in normal exposures wouldn't cause any symptoms, giving that huge amount all in one dose did cause some liver inflammation. So in the last cohort, we actually gave people steroids, just a burst of steroids for several weeks, and that controlled it. We gave it prophylactically. The LFTs did not increase, and we did not see any symptoms. So there were no side effects when you used that protocol with the steroids. And the patients did fine. There were no side effects from the medication, which was the gene therapy, aside from the liver issues, which were controllable with steroids. And now we have all 11 patients that received the gene therapy enrolled in a 6-year extension rollover trial. We're just following along every 6 months, seeing how they're doing. Are they still off of medication? Are they still able to eat protein? And of the 7 of 11 patients who did respond and have either went completely off of their medication and have normal diets, or at least had a 50% reduction in how much of the ammonia scavengers they needed to take, and at least a 50% increase in their protein intake, their response has been durable. So the first patient that was diagnosed was 6 years ago. 6 years out, they still are off of medication and they're still eating a normal diet, and they couldn't do that before. And that's actually a male. It's a man who had a milder form of OTC deficiency. So it's really very effective, apparently. So that was convincing enough that now we are having at UH the phase 3 trial. And there's many more sites involved in the phase 3 trial. We're using that highest dose that we found was effective and safe. And the phase 3 trial, the goal is to have 20 sites worldwide. We are one of them. We're the only site in the US that's open, and we were the first ones open. We're the only ones who have actually dosed a patient with gene therapy, or it could have been placebo. So the way that this works is this is a randomized control study now. So the goal is to get 50 patients enrolled worldwide. So that's why there's 20 sites all over the globe. And what happens is there is a cohort that gets gene therapy first and steroids. And then at 6 months, they get saline and a pill that's just sugar. So it's just a placebo, but they look exactly the same. And then the other group gets the placebo first, but 6 months later we'll get gene therapy. So everyone will get gene therapy by the end of the study, but it's blinded. So our patient that we dosed in January, she might have gotten gene therapy, or maybe she got placebo, but in June, if she didn't get it the first time, she will get it the second time around. And this is for children too, so for kids 12 and up. The first phase 1/2 trial was 18 and over. We can dose patients as young as 12. So we have a 12-year-old that's coming next week for screening and probably will be eligible. So we may be the first to dose someone under the age of 18. But the goal of the phase 3 study, of course, is this is the last step before getting this FDA approved. So after we get through the phase 3 trial and we have data that indicates that this is effective and safe, it will be put to the FDA to approve this as something that any metabolic doctor could then prescribe for their patients to come in for this one-time dose. So it's really exciting. And then because we have this experience with gene therapy now, and we have a lot of PKU patients, we were approached by Sanofi to be a site for gene therapy for PKU, which is phenylketonuria, and it's caused by a deficiency in phenylalanine hydroxylase. And it actually uses the same viral vector as we're using for the OTC trial. It's AAV8, and it's targeted to the liver. And since we're not active quite yet, I can't say as much about this protocol. But basically, for those who don't know what PKU is, it's a deficiency in phenylalanine hydroxylase, an enzyme that the body makes. And what it's supposed to do is convert phenylalanine into tyrosine. And if you do not have a functional PAH gene, you accumulate lots of phenylalanine, don't make enough tyrosine. And phenylalanine itself is neurotoxic, can damage the brain. Not having enough tyrosine is a problem too, because you need that to make neurotransmitters. So your brain signaling does not work correctly if you do not have enough tyrosine. And this is actually one of the more common genetic conditions. The most common metabolic conditions is 1 in 10,000 people. It is on newborn screening now, but before it was on newborn screening, when patients were diagnosed, they were toddlers and delayed, and the damage had already been done. So the damage to the brain is irreversible, and it can lead to severe intellectual disabilities. Adults who were not diagnosed as back in the past now live in group homes, and they cannot take care of themselves. But if you intervene within the first few weeks of life, restrict phenylalanine in the diet, you can have a completely normal outcome. So it's a very effective tool to use newborn screening, but it is hard for a lifetime to have a diet that's different than everybody else and not be able to have much protein, have to take specialized formulas that don't have phenylalanine but have extra tyrosine. They don't taste great. Some people, you can give a vitamin that's a cofactor. It's tetrahydrobiopterin. It's Kuvion is the brand name. About half of people will have a better, more effective breakdown of phenylalanine with that. It may not be perfect. And there is a medication that's an injection called Palynziq that breaks down phenylalanine for you. But the issue with that is there's kind of a high rate of allergic reactions and even anaphylaxis, and it's a daily injection. So the treatment options are there, but they're not perfect. And gene therapy would also be a one-time dose, and the goal would be to cure these people with one dose. And then after that, they could have a normal diet. For the women who have PKU during their pregnancies, it's difficult to keep your phenylalanine levels under control. If the phenylalanine is high, it can damage the baby's brain, even if they don't have PKU. So if these women had an option of gene therapy before having children, it could make it much easier to have a healthy child without having to have weekly monitoring of phenylalanine and being on formula. So that is very exciting. And I think gene therapy, it's just kind of, it's taking off really quickly, because once we figured out how to get a working copy of a gene into the liver, you could put whatever gene you want in there. And really, we're starting with the more common metabolic conditions. But as time goes on, it'll be available for more conditions, things that are more rare than this. And it's a really exciting time to be in this field, to be able to offer potential cures to people. So that is it. Thank you.
Perspective by Tracy Marie Greenberg, who serves on the Patient Family Advisory Board at Metro Health, as well as the chair of the ADA in the city of Lakewood. Thanks, Tracy.
Hi, I'm Tracy Marie Greenberg, and I just wanted to first thank Pam and the Judge family for everything that they're doing every year for Rare Disease Day. And this has been so far an amazing day of information. I am a patient with a rare disease. You've heard a lot about metabolic disorders today and rare genetic diseases, and I have MPS, which is mucopolysaccharidosis, and I have type 4A, which is called Morquio syndrome. So I have a missing enzyme, or a lysosomal storage disorder, so I'm not storing the enzyme needed to clean out my cells from those proteins that build up when you use energy. And I just wanted to talk a few minutes about that and let you know that that's my disease. And I'm kind of the poster child for rare disease care disparities. So that's what I'm going to talk about for the next few minutes. And hopefully they have my first slide up for you to see. So basically, there is a lot of disparities when it comes to rare diseases. And the number one thing is, you can go ahead to the next slide. The number one thing is diagnosing a rare disease. So I was I actually fell into all of these categories that are listed here at one time or another. The first one is undiagnosed. So I spent a lot of my life with just a mystery disease that doctors knew I had a mystery disease, but they said, you have joint deformities and spine deformities. And so they kind of just diagnosed me a la carte for each of those symptoms. So I fell under that category, which also would be undiagnosed, underdiagnosed. So you're just being, symptomatically diagnosed, not necessarily the underlying disease. And then misdiagnosed. I was misdiagnosed. I went to geneticist, and they said genetic department. Talked to several people, and they said they thought that I had SED, which is called spondyloepiphyseal dysplasia, which basically is how I present, because I have my disease is what's called, it's basically not as severe. So it didn't appear as the classic type. Most people that have Morquio syndrome are very, very tiny. Some of them are 3 feet tall, and I am 4'9", so they didn't automatically recognize that as Morquio syndrome. And then I had a late diagnosis because I recognized that my symptoms went beyond what that disease that I had been misdiagnosed with was, and I started doing research on my own and was able to go to a children's hospital to get diagnosed at age 42. So I've only been diagnosed with Morquio syndrome for 5 years. And now I am receiving enzyme replacement therapies on a weekly basis. I apologize that I couldn't be there today in person. Actually, yesterday, my infusion nurse wasn't able to get a vein on me, and so I'm getting my infusion today instead. Okay, and so we'll go ahead to the next slide. And I just wanted to say, in regards to underdiagnosis or lack of being able to be diagnosed, is one of the top disparities in rare disease care. Okay, so then there's a number of things that I personally experience with the care of my disease that is lacking. Obviously, the first one is going to be being misdiagnosed, underdiagnosed, undiagnosed. But the clinical setting is not set up for rare disease care. I personally experienced that this year. Last year in November, I had major surgery, and they have a lot of protocols. If you go have a hip replacement and you have diabetes, or you have heart disease, or you have any of these common things that have protocols, they have a protocol for you. But if you have a rare disease, a lot of times you can't even get your enzyme replacement therapies in the hospital if you're not already doing it through that hospital because of the expense. There's a lot of logistics involved. They will not allow your home nurse to bring in your home medication in the hospital setting because of legalities or whatever reasons. But and then in order to get the enzyme replacement therapy, if I was an inpatient, I would have to be the one to call the departments that handle that to set it up and have my home medication delivered, possibly to get that in the hospital. Nurses would have had to been trained, and it's not that complicated of a treatment. So it's a big disparity for people with rare diseases that go into inpatient care, especially adults. Children in the pediatric setting, there's a lot more set up for the children to be able to get those specialty treatments. But once you get into adulthood, it's a little bit of a different story. And then the lack of training, obviously, in rare disease care. You can only be specialized in so many things. And lack of accessibility due to barriers. This is all like the same thing that I talked about with not being able to get a specialty medication. My medication is $20,000 a week, and that's for life. So some of these genetic treatments, gene editing, and all the things that were mentioned before me I'm really looking forward to hopefully being able to join in on that and possibly be part of a trial or get that treatment to be able to not have to get a 6.5-hour IV treatment every week. Okay, so we'll go to the next slide. And now I don't want to just talk about grievances and problems because, we're here to talk about solutions. And while I'm talking about the disparities of rare disease care, we could also talk about how we can solve some of these problems. And obviously we've talked about today how we can solve it by just curing the rare disease, and also doing newborn screenings for a lot of these diseases that aren't currently on the list. Even though they are incredibly rare, especially the autosomal recessive genetic disorders, you wouldn't necessarily know that the parents were carriers of that disease until, actually for me, it was age 3 that they found out that I hadn't even anything wrong, because I was probably receiving enzymes in utero from my mom, and then it took, a while for my development to show that I was not developing normally as a child. I also have a brother who has Morquio syndrome, and he also went undiagnosed just as long as I did. And so that's how they knew it was genetic. So basically, I've always hoped to see Cleveland have a rare disease center. I think that having a rare disease center encompasses some of the complexities that come along with each individual rare disease. And if a doctor or a practitioner specializes in rare diseases, then they have a more broad knowledge of how to diagnose a mystery disease, so to speak. And also having rare disease care as a specialty. I didn't know till today, I didn't know that there was metabolic specialists, and I would certainly love to meet one and be treated by one. I get my enzyme replacements prescribed by my geneticist. So nobody's testing me for anything to see if my enzymes have gone up, and I'd really like to do that. When I first got diagnosed, I told my primary care doctor that I have this disease, and I was basically told that she doesn't specialize in it and she doesn't know how she can help me. So we talked about the inpatient comprehensive care need, in pediatric setting. Obviously you're dealing with kids, they're going to get more hands-on attention. We do all this effort to try to help people with rare diseases survive into adulthood. But once you get over that threshold, there's a lot of lack of understanding on how the damage that's already been done is going to be treated for patients that survive into adulthood and older care. And then just closely evaluating the accessibility to not just patients, but to your students, your doctors, anybody in the hospital setting, anybody in a caregiver setting, to give them access to the tools that they need to understand rare diseases and to identify, what can be done from their end to help with the patient that's being treated for the rare disease. And go ahead to the next slide. And then I just wanted to mention, when I was trying to get diagnosed, even though I knew I had a rare disease, I didn't know what it was. I would always be told, when you hear the sound, doctors are taught when you hear the sound of hoofbeats, they're told to think horses, not zebras. And I didn't know what that meant, but now I do. It's just that they're trying to think of the most obvious cause of any given symptom. And that, I think, I don't like it that they teach that, because if we're not diagnosed, then why are we teaching our medical students to not think of rare diseases? So it could be zebras, and it often is zebras. So why not zebras? And again, that's it for me. And thank you very, very much for having me. And I really appreciate all the hard work that all the researchers and doctors are doing to try to make things better for those of us who are rare.
Good morning. I am Corey Wolf. I'm so honored to share my thoughts on the caregiver perspective with you today. I'm so thankful for the invitation from Pam Judge and for the work of this whole committee to put the day together. Thank you for including me. I'm a rare mom and an advocate, not only for my daughter Meg, who lives with neuromyelitis optica spectrum disorder, but also for the NMO community through my work as patient advocacy manager for the Guthrie-Jackson Charitable Foundation. I would be amiss to not share with you a brief synopsis of the work of the foundation before I share with you my caregiver perspective. Guthrie-Jackson to date has invested over $75 million into research for NMOSD. They began with a mission critical goal to fund research that accelerates the development and approval of safe and effective therapies in NMOSD. And in just over a decade, patients today have 3 approved therapies. We're incredibly thankful to the patient heroes who participate in clinical trials, to researchers who move the science forward, to healthcare providers who guide clinical care, and to our industry and regulatory partners, of course. One approved therapy that significantly reduces the risk of relapse would have been a revolutionary accomplishment in the field of NMOSD. 3 approved therapies in just 1 decade, when there have been none for over 100 years, is a revolutionary achievement in the entire field of medicine. These advances will save and improve lives around the world, and they show how a rare disease like NMOSD can be a model for solving other diseases, rare and not so rare. No wonder why the American Association of Neurology called 2019 to 2020 the year of NMO. Guthrie-Jackson is proud to have been a catalyst and played a leading role in this, and the foundation is now accelerating research towards cures. To read more about what the Guthrie-Jackson Charitable Foundation is working towards in the field of tolerization and regeneration, please visit GuthieJacksonFoundation.org. The perspective from where we sit as caregivers is often not always in tune with the perspective of a patient. Sometimes a patient is too young or the disease is too debilitating, and the caregiver perspective may be the only representation of the patient voice. Some things become clear to us before the person we are caring for even notices the things themselves. Much of the time, we as caregivers are the first ones to know that something is just not right. Our child's demeanor, their gait, tripping, the way that they are compensating for lack of vision, or their dexterity, changes in eating, swallowing, bowel and bladder habits, energy levels, eye contact, speech, grades if they're in school, and many other observations. And for those of us who are raising children who were healthy for many years prior to a diagnosis, there is a baseline that we know by the time the diagnostic journey is underway. That baseline is rarely the place that starts where the specialists come in. And they're coming in during the journey. You're already along the path. They're meeting your child for the first time while symptoms are already happening. And it's our job as the caregivers to communicate clearly what's changed, what it looks like to you. We have to keep notes. It is so many details. We have to keep track of every change. We have to stay calm and collected, which is sometimes very difficult. And we stay the course, determined to convey the most important details without talking too much, which is sometimes difficult for me, and I think a lot of other moms. It's hard to do this when all you want to do is scream, "There is something wrong with my child, and I know it, and I need you to know it too." I have found it to be imperative that I cultivate a good relationship with my child's clinicians. And I think that that's important for all caregivers. I had a conversation with a patient once who felt she was just not being heard by her doctor, and she paused the appointment and said, "We need to start over. We're going to be together for a long time. This is an incurable disease, and you are the only person at this hospital who can help treat me. And we need to have and build a good relationship because we're going to be in this together for the long haul." I loved that and respected that approach. And I put that little pearl of wisdom in my back pocket. And I remind myself to pull that out whenever necessary with new doctors. I have asked so many experts over the years, so many clinicians, how can we address you when we disagree with your thoughts and your plan? And it's a really uncomfortable question for clinicians to answer. I understand that. And for the medical students today, I would ask you to consider, how will you react when a parent or a care partner coming to you who are in the thick of a stressful and emotional diagnostic journey with their loved one, or who has already been living in a rare disease, how will you react when you're challenged? I hope you'll listen and think beyond the obvious. Consider the impact on quality of life in delaying a diagnosis or a treatment, and really hear the details a caregiver is sharing with you. I want to share with you 2 personal examples of this. My daughter Meg was diagnosed at 15, and at the start of things changing for her, she was losing weight and she wasn't able to even finish a meal. She was often nauseous, extremely fatigued. Her hair was thinning. She was beginning to have strange rashes that would come and go. Areas of her skin were burning like sunburn when she hadn't been outside in the sun. And as this progressed, she started having recurring hiccups and then intense headaches that we later understood to be trigeminal neuralgia. The headaches were so intense she would just fall to the ground screaming, but they would only last 1 or 2 minutes, and not often in the presence of her doctors or specialists. We were told at the hospital at one visit early on, she's just a teenager. Teenage girls manifest these symptoms. They want attention. It happens all the time. And, we knew this was not the case for our child. I asked Meg to make a list of all of her symptoms. And I thought I was teaching her to be an advocate for herself. So she brought this list to the next new pediatrician's visit. And the doctor said to her, "This is a long list of complaints for a 15-year-old. And you should be happy you've lost all this weight. Most teenage girls wouldn't care how they did it. They'd just be glad to be as thin as you are. You look fantastic." But I knew, my daughter knew, and my husband, Jeff, who has been a nurse with Cleveland Clinic for over 25 years, we knew that these symptoms were not stress. We knew that she was not trying to get attention. We knew our child's demeanor, and we advocated for her and made it clear that these things were new and unusual. And we finally advocated for that MRI, and it ended up showing the worst longitudinal lesions from NMO that this group of neurologists had ever seen. It's strange that getting a terrifying diagnosis felt like a victory. And then a few years into her life with NMO, I began noticing that Meg's vocabulary was strange, something that she didn't notice. Her sentences were broken, her personality was off. She, prior to having NMO, was a creative writer and very involved in musical theater. And in a matter of a few weeks, she was not able to put sentences on paper, and her memory was terribly affected. I contacted her neurologist, and he had her come in. Her exam did not present with any findings, but I knew something was very wrong. So we advocated and pushed for another MRI, and eventually it was ordered. And Meg indeed was having a relapse, and she had lesions on her brain. But the delay in treatment made for a long and difficult recovery, and she continues to have difficulty with memorizing today. The caregiver perspective can be crucial in preventing further permanent disability for those in the rare disease community. In the transition from pediatrics to adult care, please don't dismiss that care partner. You are seeing patients for an hour, every few months, maybe even less, but we live with and care for our children, our spouses, and our loved ones, and we often observe the changes first. For parents and care partners, don't be afraid to advocate. And for clinicians, please listen to the loved one in the room. Thank you for having me.
Like many new parents, Sophie and I read all the parenting books, and we were ready to take on that typical new baby challenge. However, we quickly went from thriving to surviving. We were worried, sleep deprived, and desperate for help. We do know Cooper is challenged with hypotonia, esotropia, strabismus, autism spectrum disorder, chronic ear infections, and global development delays. He is also being diagnosed with several painful autoimmune diseases, including HSP, EOE, and EGE.
In 2019, we were introduced to the Harrington Discovery Institute. We finally felt one thing we didn't have, hope. And on that, my family all flew to Cleveland, Ohio for an intense visit. Dr. Stamler and his team of physician scientists had done their homework from all the medical information Sophie and Jordan provided. They described their process and the possibilities for an FBXO11 cure. This could result in a spectrum of positive outcomes, improving Cooper's developmental abilities and his quality of life. This process, this cure, which they believe is technologically at hand, if it is successful, it could help cure a myriad of genetic, neurologic, developmental disorders for millions of people, just like my Cooper. It would be an incredible medical breakthrough.
Harrington Discovery Institute turned on the lights in our blacked out world. I ask on behalf of Cooper's Voice, please help fuel Harrington Discovery Institute's ability to change this world for the better and impact the lives of those that cannot help themselves.
All right, hello and welcome everyone. It's my pleasure. Perfect. We have an intro slide. So Justin Abutamarco, I am a clinician over at the Cleveland Clinic Mellon Center, and it is truly an honor to be here, especially with such a wide group of people here. And I'm going to be giving this talk on NMOSD. I had Corey give a perfect introduction to this kind of topic. And we're going to be talking about the challenges and opportunities that come with this. I have a couple of disclosures. Some include some of the pharmaceutical companies that make the medications for NMOSD, but we won't be talking about anything in particular. And I really wanted to take us on a little bit of a journey. Again, we have this wide breadth of audience, caregivers, patients, people outside the medical community. And so I don't want to dive too much into what NMOSD is, but how it serves as this blueprint, right, for other rare diseases. And then what happens when we have successful treatments out there? We've touched on some of these challenges, but we'll use this kind of journey as this example to kind of go through this. We'll talk about a patient's experience, and we'll shape up at the end. So NMOSD in the real world, this was a 23-year-old young woman who came to see me last year. She's lived in Cleveland her whole life, healthy, no contact with the medical community before this. And about 2 weeks before she goes to the hospital, she's having a hard time with some higher level activities, going up the stairs, doing her exercise routine that she's always done. Didn't think much of it, maybe thought she had a little bit of a cold, but those symptoms progress to the point where she's having a hard time walking around her apartment. Severe pain kind of radiates from the back into her front. And what really scares is that she loses control of her bladder. And so she goes to Ahuja Medical Center with our UH colleagues, immediately admitted, gets this MRI where we see this severe inflammation of that spinal cord that Corey kind of mentioned, this myelitis. They do a wonderful job where they get the right blood test, they make this diagnosis of NMOSD, and she's discharged from the hospital after the correct treatment and comes to see us at the Mellon Center. And so we'll just do a quick kind of recap on what this disease is, because I know we have people from other fields. So this is an autoimmune disease. So we have the body's immune system is overactive, and it's attacking primarily the eye. So this optic nerve, where the information from the globe is then processed into the back of the brain, or the spinal cord, where all this kind of information from the brain to the extremities, to your bladder, all of that's all kind of combining into this one small area. And so these are the two areas where this antibody, AQP4, aquaporin 4 antibodies, seems to mediate the severe inflammation. So this is the kind of disease that we're talking about here. And how does it look like in the clinic, right? So when we're dealing with the eye, this optic nerve, people could have vision loss in one eye or even both eyes during their first attack. It can be absolutely devastating. And then with our patients' description or Corey's description, where we hear about what it can look like when it affects the spinal cord. Difficulty walking, pain, And in even rarer cases, we can have these hiccups or nausea or vomiting that Corey mentioned, which is even more difficult to kind of put together. It's not a typical symptom we think of with the brain. So this is how this disease can look like. And what we know is that NMOSD is going to be a relapsing disease. It's going to happen over and over again unless we treat it, unless we find out what's going on. But one attack can leave people with severe disability. One attack, people can be left blind in one eye or both eyes. They can be left with the inability to walk. So these are some outcomes just at 5 years that, you know, the majority of patients will be requiring a wheelchair, will need some visual assistance because of the deficits there. So there's this really devastating disease course. So I said we go on a little bit of a journey. So we're going to go back to the 1800s, okay? We have Eugene Devic. Eugene was living in France, middle-class kind of community. His father was a wine merchant. I was hoping he would go into the family business, but instead he went to medicine. And by all accounts was a wonderful clinician, a wonderful teacher, and had a great bedside manner. And he kind of put together this disease with him and his students. He was able to kind of piece together this idea that the inflammation that was involving the eye for some of his patients was also affecting the spinal cord. And that seems intuitive now, but that was not the case back then. And so Eugene Devic was one of the first descriptors of this neuromyelitis optica disease. And it was originally described as Devic's disease before this. But we went over 100 years, right? Corey kind of mentioned this. We thought this was a severe variant of MS. We couldn't differentiate it, though I think we all knew in our gut that it seemed to be something different. 2004, we have this incredible discovery of this aquaporin 4 antibody, right? We can finally differentiate it from MS. We know what's causing it, and we have an easy blood test to kind of differentiate that. And so we have this revolution in the field. And we really quickly moved when the field came together with the pharmaceutical companies, patients, clinicians, to develop diagnostic criteria so we could make the diagnosis. And then we were able to develop our first FDA trials, clinical trials in 2015, and our first FDA-approved treatments in 2019. And for those who maybe are not a part of the medical community, right, this seems like a terribly long timeline, right? We go from 1800s to 2023, right, where we're starting to get these therapies available to patients. But this discovery from 2004 on to discover a disease and then develop multiple therapies is incredible. And it shows what we can do when we all come together for these types of diseases. So we have these three options, a fourth probably on its way. The names aren't important, but you know, we have multiple options where we can talk about patients about what fits best with their lifestyle, right? Infusions, injections, whatever works best for them, it's incredible. I'm glad most of you guys are seated because the next slide's a little tough, right? The cost. And we've been hinting at this throughout the talk, but the numbers are staggering, right? $200,000, $400,000, $700,000 a year. It's not a one-time treatment. It's not a 5-year treatment. It's a lifelong treatment, right? How do we get access to these medications for everyone that needs them? So let's go back to our case. So this young woman who's seeing us, she comes in and she has this kind of wonderful disposition about her. And she comes in, she's like, she knows what's going on and she has a plan. And she tells me, she's like, I'm leaving in 4 weeks. I'm going to graduate school. She had already had plans to go to medical school after this. And she's like, whatever we're going to do, we're going to work around this. And I said, okay. So we got a group of people around her to make sure that we could get her better, both from a physical and mental standpoint, right? This disease requires a whole community to come together in order to treat and to manage this disease. This is one of the pictures we kind of put together for another talk, but you know, when we treat these kind of patients, and I think this applies for all of these rare diseases, right? The patient has to be at the center. Maybe someone kind of quarterbacking all the different groups that need to be involved, but we need to have our rehabilitation experts, mental health experts, patient advocacy needs to be a part of that, and we need to make sure that we're empowering patients. And so this is the kind of diagram that I like to think about when I'm seeing patients, and we were able to get her connected with those different groups. But there's something outside of the clinic, something that we can't touch and feel sometimes, but we know is impacting care. And we've kind of termed these as social determinants of health. It's a fancy word to say what's going on outside of that clinic visit. What's their home life like? What's their social and community context? Their financial stability? What kind of education background do they have? These rare diseases are hard to study in this kind of context. We're actually starting one at the Cleveland Clinic, but we have good data in multiple sclerosis showing that these impact outcomes in the long term, right? That's not surprising, but we haven't seen that as much in the literature until recently. And in particular, African American patients can be particularly affected by these. And so how do we address these pieces? I know in this audience, right, we don't have to talk about this very much, but the social inequalities woven into our society are tremendous here. So here are two diagrams. That top one is this index kind of value so that we can study it a little bit easier, but it combines people's housing, financial stability, education background into a number. And those darker numbers aren't good. Those darker numbers mean higher socioeconomic kind of disadvantage. And we know that it interfaces with our kind of health outcomes. So mortality here is listed in that red slide, where those darker colors kind of show poor outcomes, and they're directly linked, right? But how do we address these in the clinic? So when we look at NMOSD, right? Corey's example, her daughter was 15. This disease affects young people. So we're talking about average age of onset is around 40, but we see patients in their pediatric years, so under 18, 20s, and 30s. The prime of their lives, right? Our patient, she was trying to go to school, family planning, career planning. We're right in the thick of that. It affects mostly non-white, non-Caucasian patients. And then females are affected much greater than males, about a 10-to-1 ratio. So we have all these factors kind of coming together, I think, making a really challenging kind of disease to treat and manage. This is one of my favorite kind of figures, and I think for the patients and caregivers that are in the room, I think it resonates with how their experience has been within the medical community. For those outside of it, I think we expect when you go to the hospital, right, that treatment trajectory is just going to be this straight line. It very rarely is, right? These ups and downs, and I try to set that stage, but I think it feels more like that second kind of line all over the place, right? The emotional kind of ups and downs of receiving a big diagnosis. But we do have a little bit of a success story with this young patient. You know, we were able to decide on starting her on infusion-based therapy that fit with her lifestyle, right? It was infused once every 6 months, and we were able to make those infusions around her school breaks. So she was able to come back to Cleveland and make sure that worked with her lifestyle. We were able to get her connected with that multidisciplinary team, PT, OT, speech therapy, our physical medicine rehabilitation experts to get her feeling better, and our mental health experts as well. And she was able to make a full recovery, and we were able to get her on copay assistance so that she didn't have any out-of-pocket expenses, so that didn't have to be another barrier to her receiving care. But these stories don't always end this way. The patient advocacy organizations, I've kind of touched on it, it's been touched on by a couple others, but are so important. I've had the pleasure of working with a few here, and the Connor B. Judge Foundation has just been an incredible resource, but not only to connect patients, to have a community outside of the clinic, but the financial support that they provide is something that just can't be highlighted. And so we have to continue to support these kind of groups. So in conclusion, I hope that I've done a good job using NMOSD as this example, this blueprint for how we can tackle rare diseases together, right? We can have success in a relatively short period of time. This multidisciplinary care model where we have multiple people coming together in order to help patients is so important. And we have challenges, not only inside the clinic, but outside the clinic that we have to think about, and we're not there yet. I think having some increased awareness will help, but we have a long way to go. I'm from New Jersey, so I think I can share this slide. It's also one of my favorite cartoons. Whenever I have a patient encounter, right, when they leave my office, I try to tell them, like, I want you to see that light at the end of the tunnel. They don't always feel that way, but I try to have them, I want some hope, right? Whether it's a diagnosis, a potential treatment, this kind of multidisciplinary approach that we'll have. And so this is a cartoon that says, discovering that the light at the end of the tunnel is in New Jersey, and he's not very happy about it, which I understand. It's why I'm in Cleveland. But yeah, it's with that idea that we'll end. And yeah, it's been a pleasure. Thank you.
Hi everyone, my name is Urvi Gupta and I'm currently a second-year medical student at Case Western. Thank you so much for this opportunity to speak with you today. It's really amazing to get to talk about my experience as a medical student interested in rare disease education. It's something we don't get to talk a lot about at school, so I'm really excited to share this with you. So just an overview. So I'll talk a bit about my background, my previous experience with rare diseases, and then I'll talk about my main source of exposure to the rare disease community, which was through the Rare Compassion Program. I'll talk about the lessons that I learned through the program, and finally I'll talk about some ways to get involved. So like I said, I'm a current second year at Case. I actually went to Case for undergrad as well, so I basically lived in this building actually for years. And entering medical school, I'd never really heard much about rare diseases. I'd never, encountered any patients with them, and it wasn't actually until around halfway through my first year that I just happened to hear about rare diseases. We have these optional lectures where patients come in and talk to us, and the patient for this one lecture has Fragile X. And she was talking, she just mentioned that she was involved in this program called the Rare Compassion Program. And I'd never heard of it, so I went home and I looked it up. And I could not be more thankful for that one experience because it has really opened my eyes to the world of rare diseases. And I just want to emphasize that, this is not something that was widely advertised. It was really a unique experience, and one of the reasons that I really want to advocate for increased medical education on rare diseases. So how the Rare Compassion Program works is medical students are paired with rare disease patients and their families. So over the course of 8 months, we're paired with 4 different patient partners. We meet with these patients over Zoom, and we just talk. We just learn about, their diagnostic journeys. We learn about how they're currently navigating their treatment, the impacts it has on their families and their friends, and also just, their hobbies and what kinds of things they like to do, and who they are beyond their disease. And as a medical student, this was the first time I really got to just sit with a patient and talk to them. And it really taught me so much about medicine in general, but also, rare disease patient care. And those are some of the lessons that I want to share with you in the next slide. And another element of this program was these end of rotation meetings that we had. So at the end of every 2 months, we had meetings where all of the medical students would gather together. And we just talked about the different lessons that we learned from our patient partners. We had one really interesting session where we actually watched an example of, a negative interaction with a patient and a doctor where the doctor was really not listening to the patient, was kind of talking over them, was making assumptions, similar to the ones that we heard our earlier speakers talking about that doctors often made about them. And then we followed that up by watching the exact same interaction, but with the doctor actually, really listening and showing that they were actively listening to what the patient was talking about and demonstrating that partnership. So overall, the program was such a great way to just learn about these themes that, we don't really get much exposure to. So some of the lessons that I learned, I had never known that, anything really about rare disease patients. So through this program, I learned about all these special considerations to take into account. I had never known that patients often go, decades without having a diagnosis. And it was really interesting. Even our earlier speakers mentioned that, even though it can be a devastating diagnosis, it does feel like a victory to receive one. And that's not something I'd ever understood or been exposed to through the curriculum. One of my patient partners, her and both of her kids have Loeys-Dietz syndrome, which was a connective tissue disorder, which can cause all kinds of symptoms and manifestations. So one of the things she talked to me a lot about was her healthcare anxiety. Every symptom that she experienced or one of her kids experienced, they were concerned that it was a rare manifestation of their disease because not much is known about it. And so, you know, seeing how things like COVID and, you know, other things can really impact a family with a rare disease was really important to me. And, you know, something that is a special consideration for rare patients. And the impact on family and personal life, you know, having to navigate all of the appointments and the insurance landscape and navigating goals of care. You know, these are things that I really had never known, and getting to hear my patient partners talk about this was so important in shaping how I am going to interact with my future rare patients. And the importance of a strong doctor-patient relationship. You know, sometimes just having a doctor say, you know, I don't know at this stage what your diagnosis is, but I will work with you and we'll work together until we find it. And, you know, that was oftentimes all patients needed to hear at that moment in time, just knowing that someone is there to be next to them in their journey. And that patient earlier, the one who had a lot of healthcare anxiety about her and her kids' symptoms, you know, she said that having an open communication with her doctor made her feel so much more comfort with dealing with that healthcare anxiety, because she knew that she could bring up these symptoms she was experiencing without, you know, seeming overprotective, or in her words, she said she was worried about seeming like a helicopter parent. But, you know, this doctor really understood what she was going through and was able to work with her and really investigate all of her symptoms. And just the importance of shared decision making. We just heard about how, you know, there can be different treatment options and different things to really take into account when navigating, designing a treatment plan. So learning how doctors and patients have to work together to, you know, meet at common goals. That's something that not all, you know, patients with common conditions, often there's a very set rule book, you know, but patients with rare diseases don't often have that. So the importance of shared decision making is just that much more essential. And getting involved, medical students and patient partners, the Rare Compassion Program, again, I can't emphasize enough how great of an opportunity that was, and they're always looking for more people. So if you're interested at all, please look into that. Something else, the Ohio State Resource Project, it's a state initiative where they're compiling online resources for rare patients. So any sort of information regarding, you know, insurance information or how to find specialists in your area. It's an effort to bring all those resources into one place. So that's something that anyone can get involved with. And something I just learned about yesterday actually was there's a group of medical students at a couple different schools who are trying to put together rare disease interest groups at all different medical schools. So that's something I'm definitely interested in. And if anyone else is interested or has thoughts on that, I'd love to hear them. But yeah, thank you so much for this opportunity. I have my email up here. Please feel free to reach out with anything. I would love to connect. And yeah, thank you so much for your time today.
Okay, hi everybody, I'm Dr. Mary Rensel. Thank you all the speakers that are ahead of me. I love this day because I learned so much. All right, so let's see, hopefully we get the slides. All right, we're going to talk about a framework. We are all advocates in one way or another. So let's learn a few tools or a few tactics that we can use so that we can be even stronger advocates and get our voices heard. Mrs. Wolf had shared her experience, and you know, I have a similar, but similar in the sense that I was learning how to be an advocate in a new arena when I was dealing with policymakers. I was learning how to be an advocate to our Congress and Senate and state reps. And so that was a whole new area because I had to speak their language and I had to speak fast, and I couldn't use what I knew. I couldn't tell them everything I knew about NMO or any other disease, but I had to think of what they wanted and needed so that they could share with their teams what was important to them. So when we are looking for a high success rate as advocates, we want to package our message like a gift to the people that we're talking to so they don't have to work hard to understand it. We want to present it in such a way that it's right there, there's a nice little bow on it, and they understand the information. So I had to learn how to do that, and it took a while to learn how to do that when I was interacting with our policymakers. So we're going to talk about when we are advocates, how can we focus on the audience? Because it's not about us. It's about the audience that we're trying to share the message with, like our med student who just rocked her presentation, telling us what it was like when she was interacting with those families. I felt like I was there as part of that conversation, the way she packaged that for us. And now we know exactly where to go. She shared the stories, and we know exactly where to go if we want more information for that. So we want to give people a framework, like this is the quick message. This is how we want to give the information in a really understandable way, give them a framework so that it helps that person. So most of us are sitting here thinking like, I need something, so when we are sharing a message, I always want to be thinking of you, not me. It's not about me today. It's about you, what you need to be better advocates, because I feel like there's still a lot of opportunities in this world to make this world a better place for so many people living with rare diseases and many other issues as well, right? So if there's anything I can do to serve you so that you can go out and be really powerful advocates, I know it's just going to be a ripple effect around the world. So it's not about me as the speaker. So when we are advocates, we don't want to think about what we need. We want to think about what our audience needs. So where do we start? So an easy method when we're giving a presentation is something called the Minto method. So we start with the answer. And the answer today is that you all are wonderful advocates, and we have a framework that we can use to be even better advocates. And then where do we start? Well, we think of our audience. We think about serving them. So we want to give the answer first when we get up and give a talk so that everyone knows why we're there. So we start with our key takeaway. We give them 3 points. We can have 3 points of our 3 points, but think threes. People really, brains like threes. And so you want to just say, okay, that's all I can give you. You want to tell them everything. You want to show, oh, you had so many experiences. There are probably so many amazing conversations that, the med student had, with her families, but she just chose a few. So you just want to choose a few to highlight the really important things to get, again, think of that bow or that package you're giving so that people can be better advocates and take it back to their team. So you're amazing. You need to know yourself. You need to know your gifts and your strengths that will make you a special advocate. And we've heard from researchers, from moms, from med students. So you can be an advocate from so many different angles. And we can all follow this framework. So we've had a lot of sitting today. We've had a lot of listening. So we're going to do a little exercise. Pam said, make it fun. So I'm listening to Pam. You know me. I always listen to Pam. I've known her a lot of years. Okay, so let's do this. So, a lot of times when you're sitting there, if I know why you're there, it means the world to me, right? I know what's important to you. If I knew your purpose, why you're here today, I probably need so many Kleenexes because I'm sure you all have amazing stories. So let's work on a little exercise that helps us share our purpose. So think you're at a dinner party. Hey, what do you do? And I pick on Dr. Abatamarco. We work together, so I get to pick on him. What do you do? He might say, I'm a neurologist. I'm a neuroimmunologist. I work at the Mellon Center. I specialize in autoimmune disease. But what if he started with the last question up here? So I'm going to ask you to turn to the person closest to you at your table and answer this question, the last question. How do folks feel after they interact with you, whether it be work or your advocacy work? How do they feel? So I work at the Mellon Center. I see patients. I'm hoping, and caregivers, I'm hoping that they feel heard, valued, informed, empowered. I'm hoping they feel supported. So I'm going to give you a minute. Turn to your person next to you. What are the feelings that people feel when you're advocating, or if it's your work? I see a lot of students here today, so you guys, I'm sure, already know the answers. So how do folks feel? You get 1 minute. Hi, Tessa. Okay, we're going to come back together because Pam also told me I only had 5 minutes. So, okay, I know you have a lot of ideas, big ideas. So you're at a dinner party and someone says, hey, what do you do? I'm going to pick on this table here because I don't know you. But so they're going to say, hey, I help people feel. What are some words you came up with?
So we're from Harrington Discovery Institute.
Nice.
And we said that we hope that people feel
educated, informed, inspired. They're from Harrington Institute. Nice. Okay. So if I was there, I was sitting at, like a wedding kind of thing or something, and you sit down and what do you do? Well, I help people feel educated, inspired, informed at the Harrington Institute. And I'd be like, really? How do you do that? But see, what will happen is you will go reverse on these questions. And if they're interested, sometimes they're not, of course they will be. But then you say, well, how do you do it? Why do you do it? How, what does your day look like? So then you can go from there. So that's if you share your purpose, you're going to link, at that deep emotional level pretty quickly. Some people will be interested, some people will not be. But, that's one way to know if someone is kind of on that wavelength with you. Are they synchronizing? They care. They want to be inspired. They want to know. They want to know more. And when they go home, they're going to remember you. They're going to remember that, right? So that's a deeper level saying, oh, I'm a banker, I'm a teller, I'm a doc, I'm a nurse, I'm a researcher. And they're like, oh, that's cool. Because if they don't know anything about researcher, then where do they start asking questions? How do they get to know more? They might think like they didn't want to look like they don't understand research, So if you say, I help people feel inspired, educated, they say, oh, how do you do that? It really kind of opens the door to the issue. Because again, we're trying to advocate. We're trying to share our purpose and our mission to make this world a better place for whatever disease state or whatever, your mission is on this world. So that is one way to be very clear about your purpose. And then you can use it in different situations, and when you're at, it could be, dinner party, wedding, networking, what have you, sports events. And people can tell then you're just there to help others. You're there to support others. And it really, it will just open the door to more conversation. So hopefully then you get to share like, hey, we're part of NORD, we're part of this. Come see us. We do research. We have a fundraiser next week. Come on into our fundraiser. So it just helps to get the conversation going as an advocate so you can open the door. Okay? So when you get home, write something down. Start with the feelings and then do it in reverse order, because really our purpose is to lift others that are suffering, to take care of the areas in this world that we need more support. Maybe we need more funding for more research. We need people, we need boots on the ground, we need providers, whatever we need to let people know what's important to us so they want to join our team as advocates. Okay, so you got this. So get out there and share your message, share your purpose, like a bow, like with a big, a gift with a big bow on it, so that people know exactly what you're about. So when they do have an idea, if they have, they won the lottery, they won this thing, who was that person I met at that wedding that did X, Y, and Z? I'm going to go ask them, if they're having a fundraiser or something. So you'd be surprised, if you hit people at that emotional level, it'll lead to more questions. It'll give you a framework to share your advocacy mission. Thank you. That's it.
Good morning. Thanks for having me here today, and I'm delighted to meet some new people, reconnect with my colleagues here in Cleveland, and just try to give you today a couple of messages. I'm going to try and follow Mary's recommendation today. I'll give you the pitch in the beginning, and then we can go over details. So the two things I want to tell you is that, and I was asking myself this question: what does rare mean? We talk about rare diseases. Rare is precious. Rare is beautiful, and rare is rare in the meaning of the word, but when it comes to the individual patient, to the individual family, it's not rare. It's 100%. It's everybody in that small group, and its impact is very large. So rare can mean a number of things. I'm a pediatric ophthalmologist and geneticist. Now my practice has been restricted to genetics only going forward. I took these pictures in the Cleveland Aquarium. They were posted on the wall, and they're pictures of eyes from different species. So do you think they're rare? They're not. Every one of these is in one of billions of fishes. But I find them really beautiful and attractive. So I wanted to decide to show them to you. So rare diseases of the eye have a big impact, and they have impact not only on vision, but on life, on family givers, on the community, and on society broadly. And I'm talking about eye problems here, but that applies to any of the rare diseases that we're talking about, because they all affect one of our organ systems, if not all our organ systems, in many of them, and that will have impact on all these areas. I can't help but think of genetic diseases when I think of rare diseases, and for the most part they are, but some of them are not, immune disorders, although they are also genetically underlying. But there are also rare infectious diseases, rare disorders that do not have a genetic underpinning. But genetic eye diseases are the largest group of unavoidable but sometimes treatable blindness in developed countries. If you go to Africa, it's not going to be the genetic diseases that are going to be the most impactful on vision. It's going to be vitamin A deficiency. It's going to be trauma. It could be a number of other things. But in our own environment here, the genetic eye diseases are the most common causes of blindness. And if we take a study of a developed country like Britain, where visual disability is defined as vision that's reduced to 20/200 or less, about 1 in 1,000 persons have visual disability. Okay, and half of these cases are presented, are diagnosed or present to the eye doctor in the first year of life. And when we look to the cause of these visual disabilities, we find that a cerebral visual impairment, mostly from hypoxia and perinatal changes, retinal disorders, which is mostly genetically determined, and optic nerve disorders, which are also mostly genetically determined, are the most common. And something that Dr. Abeta-Marco touched upon with the social determinants of health, it turns out also that the incidence is higher in children from ethnic minority backgrounds, lower socioeconomic status groups, and those who are born prematurely, which also takes us back to the same groups. The other important thing is that these children, these adults, when they become adults, do not only have eye problems, they have systemic problems. They have other associated problems in other body systems. And if we take them in the first year of life and follow them up, 4% would have died within a year after a diagnosis of their visual disability. So they can have very, very serious other problems that are lethal. I'm showing you here the rarest of patients that I've seen. And this is a mom and her daughter with cryptophthalmos, or hidden eye. So their upper eyelids are very tall, wide, if you like, from top to bottom, and their lid fissure is all the way in the bottom here, and they're fused together from birth, and the eyeball is behind them, and where it attaches to the skin of the eyelid from the back, there's that little dimple. So unfortunately, they're totally blind. Fortunately, everything else in their body works well, including their brain. So it's terrible that they know what's wrong and bad in their life, but at least the rest of their health is okay. I don't know how to think about this, so you think about it. Would you rather be blind and intellectually impaired, so you don't know what's happening to you, or would you rather be blind and have everything else in your body work well and be healthy? Evidently, nobody wants to have to be in either situation. That goes without saying. So we'd like to think about genetically determined eye diseases as either being isolated, so only in the eye, or as part of a systemic problem, systemic condition. And I don't recall the exact statistic, but probably 40 to 50% of systemic diseases would have some problem in the visual system and the eye. Certainly neurological disorders and dermatologic disorders would have a lot of them. Certainly connective tissue diseases also. So there are two major categories: malformations or retinal dystrophies or degenerations. These are some examples of ocular malformations, and I'm showing you some examples here. Just microphthalmia, that little girl on the top right was born with the corneas that are opacified and has glaucoma, so her eye is enlarged. Unfortunately, she does not have any vision at all. Bottom left there, this is optic nerve hypoplasia, so the nerve just did not develop. That's the most common of the ocular malformations that exist. And bottom right is a coloboma, or an absence of part of the retina and the choroid, where the white is, the retina is absent there. So in addition to being a source of visual morbidity, some of these malformations, if we diagnose them, we can actually uncover some hidden abnormalities that would affect the lives of patients. This little boy was born with a third nerve palsy. The cranial nerve number 3 innervates the extraocular muscles in the eyelid. So in this patient, it was just not working at all. It was actually absent. And so his eyelid is droopy from birth, and his eye is deviated from birth. And in his retina, he had a malformation, actually, an optic nerve malformation called the morning glory disc, which you see there in that picture on the top right of the screen. And because we know that this can be associated with abnormalities of the blood vessels in the brain, he had an angiogram, and his carotid artery on the same side of that optic nerve malformation was absent. And you can see that in the bottom right there, with the carotid artery on the right side, but absent on the left side where that arrow is. So sometimes we can save these patients' lives, or anticipate at least problems that they may have because of these other malformations elsewhere in the body, including the brain. I have a special interest in early onset childhood retinal dystrophies. These are present in the first 7 years of life, and patients present with a variety of symptoms that could be nystagmus, or abnormal eye movement, shaking of the eye, vision loss, light sensitivity, night blindness, and then we can see some changes on the retina when we examine them. And there is a pathway we follow when we work up these patients. We have to ask the correct question. We have to determine whether they are isolated or whether they are in the context of a systemic disease, which is always an important thing to unravel, because they may have renal problems, they may have brain problems, and unless we make that diagnosis, we're not going to be able to involve the rest of the healthcare team in taking care of them. And finally, when we want to determine exactly what gene is involved, we go to molecular testing, and we only do that in the context of good genetic counseling. This is for our genetic counseling colleagues in the room. So that is very critical to keep in mind. There have been a lot of advances in discovering genes for inherited retinal diseases. You can see that slope going from 1990 to the present, and I think the majority, if not all, of those genes have been identified. There are some that have been mapped, but we don't know what the exact gene is, and they're now in about 350 to 400 genes that would cause inherited retinal diseases. I see those kids in my retinal dystrophy practice. And between 2012 and 2017, we collected 119 of them. And the reason I'm showing you this slide is that we were able to get molecular testing and an accurate diagnosis in 83% of cases. If you'd asked me this question in 2010, I would have told you 0. We know that they have a disease. We know what the clinical diagnosis, but we don't know what the gene is. So this is what's happened. And this is what has led us into what I'm calling conquering inherited retinal diseases, just like many other diseases have been conquered, and you heard about some of them. And that is a collaboration between a number of groups, including patients, basic scientists, industry, you know, professional organizations, support groups like this is an important support group. And we are at a time where there's a lot of advances in technology. Medical and scientific knowledge has also expanded dramatically. So putting all this together has made for us being able to conquer some of these diseases, and has led to the availability, and I can't believe I'm saying that word, the availability of gene therapy for a retinal disease, at least until now. And Dr. Mitchell mentioned that earlier today. The gene is RPE65. It's one cause of these inherited retinal diseases, and after a lot of animal model, clinical trials, and surgical trials, we are able now to deliver the gene in a viral vector under the retina and directly give it access to the retinal pigment epithelial cells and have it produce the protein that is defective here. And these patients do see better. Before the treatment, they're unable to navigate when the lights are dim, when there's nighttime in a dark room, and after the treatment, they are able to do that. I did not bring with me the movies to show you about this, but this has been tested and shown to be actually quite effective for that. And some of these patients now are at least 5 years after treatment, and they continue to have the ability to see in dim lights and in darkness. It is very expensive. When it first became available, there was an outcry. It was $1 million. It continues to be $1 million, but insurance covers it, Medicare covers it, and that's just part of the battle to be able to do that. There's about 15 to 20 other retinal dystrophies now that are in clinical trial stages. So I'm sure that this is going to become available for other diseases. So where are we in 2023? We know a lot more about the course of diseases of these inherited retinal dystrophies. So the registries that we heard about earlier today, following patients over a long time, determining what's going to happen in the majority of them, will help determine if treatments can be given, if they are worth the risk taken when they are given, if they are worth the cost. Unfortunately, that's always a consideration that is given. So we know a lot more about that now than we did 10 years ago because of registries, because of natural history studies. We are able to make a molecular diagnosis very, very early, even in utero, evidently by prenatal diagnosis. Technology has allowed a lot of better low vision rehabilitation than we ever had. And I can see it in my patients who now have a lot of different ways to communicate, to see, to magnify, to listen, to type, etc. They might be able to use self-driving cars at one point. Who knows? Neuroprotective agents are still in the works, but I do think that there will be a time when they would be effective. Gene therapy, evidently we talked about it. Stem cell therapy is not there yet for retinal diseases, but there's a lot of work going on. A retina prosthesis has been developed, and we've implanted a dozen of them here at the Cleveland Clinic in the past few years, only to stop now because they stopped making them. They turned out not to be as effective as they were anticipated to be, so they stopped them. But they're working on newer ones, so that's going to come back. And evidently prenatal diagnosis, which has been available for genetic disorders for the longest time. So I thank you for listening, and I'll pass the baton on to the next speaker. Thank you.
Well, thank you to all the speakers for setting us up for such a terrific morning, and I'm hoping that I can pull together a few different threads with some of the information that we have to share here today. I want to thank you very much, the organizers, for giving us this opportunity to talk a little bit about what we do. But so what we hope that we will deliver, that is what we hope you will receive from our presentation today, thinking about the speaker, about who's your audience, is a little bit more information about what we do at the Harrington Discovery Institute, but also I hope that what you walk away with is a sense of understanding about how this entire community makes contributions that are important for organizations like ours to facilitate and support new drug discovery. Wrong way. Okay. I seem to have seem to be missing the beginning of the talk here. So I'm going to shorten it a little bit. Let me talk a little bit about the Harrington Drug Discovery Institute's beginning. The Institute was founded in 2012 by a generous gift from the Harrington family, and we're part of university hospitals. We're part of the research mission of university hospitals, and the idea was that, as you know, there's a huge unmet medical need in many different areas of healthcare, and in particular in rare diseases. And in this last decade, there's been a lot of technology breakthroughs and a lot of information that has led to ways of diagnosing diseases that we've seen that are rare, and knowing the genetic origins of it, and knowing how the different parts of the body's mechanisms work together to either deliver healthy ability to be present in this world and do functions, or where those things get disconnected. So we know more and more about what leads to symptoms for folks with rare diseases, and we know how to identify what those are. And in the cases where there are therapies, it's absolutely fantastic when a discovery of the diagnosis leads to an existing treatment and where there can begin to be immediate impact. But in those cases where we don't have immediate therapies, what do we do about that? And so the mission of our institute has been to identify researchers who are in academia, and often they are physician scientists who are treating patients and doing research. And they have fabulous ideas for developing a new therapy that would address an unmet medical need. But they haven't necessarily had the training that it would require to advance those therapies to a point where the FDA would allow experimentation in a clinical trial, and where commercial partners would find investment in bringing forward new therapies to be approved by the FDA. And so what we try to do is we try to bridge that gap. And we see the gap as being partly funding, because we all understand how expensive it is to support medical research and how expensive it is to fund clinical trials. But it's also know-how. And we try to fill that know-how gap in ways that I'll describe. Our history now, in 10 years of working towards this mission and towards helping to facilitate the development of new drugs, has given us the opportunity to work with 165 different research teams for 165 different potential therapies, or what we think of as drugs in the making. And we are disease agnostic in the sense that we're not focused on a single disease area. And so as a result, those 165 really represent possible new therapies in many different areas. And in the rare disease community, as you're all familiar with, we have many different categories of medical diseases that they fall into. In the Harrington Discovery Institute now, we've had many rare disease projects in neurosciences, also in metabolic diseases, in ophthalmology, and that's partly through a partnership we've had with Foundation Fighting Blindness, and also rare cancers and rare hematological conditions. So it's always been close to our hearts, so to speak. We've always been appreciative of the need for rare disease therapies. But another thing that we look for, in addition to the beginning, is how can we know to advance? And so just to encourage some of the earlier presentations learning for today, without those patient stories, we're not as inspired. And so those are critically important. But without the patient cooperation with drug discovery, we wouldn't be able to conduct clinical trials. And most importantly, without natural histories and without engagement with caregivers, we wouldn't know how to measure whether those new therapies are even working. And so all of these threads coming together today is really terrific. So now in our second decade of working towards closing some of these gaps in medical need, we organize ourselves into 4 different centers. There's the Brain Health Medicine Center within Harrington, which is primarily Alzheimer's disease, Parkinson's disease, and those kinds of neurological disorders. There's the major diseases, a lot of cancers fall into that category, and cardiovascular. We did actually run a program in COVID and new approaches to treating pandemic viral infections. And we also have our Rare Disease Center. So I'm really going to be talking about what we do within the Rare Disease Center for a few more slides. We started our rare disease program not because it was the first time we ever looked at rare diseases. We did have our ophthalmology program, and we had some of our physician scientist programs were in rare disease, but we thought we really needed to address this great and growing opportunity to meet that unmet medical need in a more systematic way. And so we started in a partnership with Takeda Pharmaceutical Company, who provided us with some funding to run our first program. And I should say that in each of those centers, we think about different programs where we put out calls for applications, and we are a transatlantic international organization. So we're finding new opportunities to support in the US, in Canada, and in the UK. And we have funded these 165 projects in all three of those areas. So in 2018, we put out a call for ideas in academia for treatments for unmet need in rare diseases. And we had initially over 250 letters of intent. So it took quite a long time, but sourcing those opportunities that we think are doable was really, really critical. And so we were rigorous in our evaluation. We used reviewers who are experts in their fields, in medicine and in discovery. And we also used experts that came from industry because we wanted their inside knowledge on how to develop drugs, how a company would develop drugs. From those 250 initial letters of intent, we selected 90 applications for a full application, reviewed those, and selected ultimately 8 projects that we supported for 2 years with funding and with know-how. So I'll get to that in a second. But I did want to say that it's a long, just like sometimes the journey to diagnosis can take a long time, the journey to having a clinical candidate for testing takes a long time too, especially when you're starting with just the first point on the map. And so in the years since 2018, so far, those 8 projects we've been able to achieve in vivo, which means in an animal model, proof of concept that the idea has merit, that we could potentially develop a drug for that type of disease with this idea. We have one of those projects estimated to go to the FDA with an investigational new drug application, which means starting clinical trials sometime this year. Another one, which we think will hopefully enter the clinic in 2024. There's been a startup company started from one of these investigators, and there's also one of the projects which has been the subject of an exclusive option to license from a pharmaceutical company. So that's actually, if we look at what it takes to advance these projects, we're pretty proud of the progress that those investigators in the program have been able to make so far. We're still tracking their progress, and we'll keep you updated in the year to come. All right, so I mentioned that we support our investigators when they're selected from grant money, but also in hands-on development expertise. And we also have a network of CROs, which are contract research organizations. These are companies that do work for hire, and they have already all the instrumentation, all the know-how, and the methodologies to conduct certain experiments that the FDA might require before they would feel that it is a safe enough risk to begin human studies. And we can connect people with those CROs, and in many cases, we actually manage the relationship and manage the contracts with the CROs on behalf of those scholars. Now, up until this time, everything that I've been talking about is philanthropic support for those investigators. They're academic investigators. They do get money from NIH and other grants. But our grant money that goes to them is also a gift. In other words, we don't take the intellectual property, and we don't control any of their patents that they might. But we do have in mind that without that commercial partner down the road to go all the way from your first clinical study to an approved drug, it's highly unlikely in our world to get a brand new approved drug and to be able to market it to the patients who actually need it. And so we also support our scholars on the business development and the business strategy side. And if we think to the presentation about the packages, that means how does an academic investigator who has a really cool idea for a drug, who we've helped to move it along the pathway, how do they attract a pharma company to say, maybe you would like to license this product and put the hundreds of millions of dollars into it that it's going to take to get it over the finish line? That means you have to think about how their language, and in the same way that some of you might be working with on an advocacy on the Hill or talking to your local representative, when you're talking to a pharma person or a venture investor, you have to know their language and you have to deliver the messaging in their terms. So we help those academics learn that language and package their messaging. We have a small staff, and some of my colleagues on the program side who help us with selecting those projects are here with me today. But my personal contribution at the Institute is to help with the advisory and support side of what we do with investigators. And to that end, we have 4 project managers on our team, and Michael Hallen is one of the project managers who is actually also our senior project manager on the rare disease portfolio side of things. And so he's become incredibly educated about all of these different threads and how they come together as we try to serve the community with supporting these projects. Well, the heart of what we do and what's different about us compared to other granting organizations is that we have a large set of collaborators who have spent their careers in pharma, and they learned on the job all those things that don't get published about how you develop drugs. And if you look at the little circle on the bottom right, if you're the investigator and we've selected your project for support, you're the little gray figure in the middle. But what we do is we surround you with a lot of people who can provide their know-how and their knowledge on what might be needed to take that idea and move it all the way to a clinical candidate. So, for example, you might be someone who's working in a rare disease where you have developed an assay and you might have a model that you're testing it in your lab, but maybe you want to do a toxicology study, and we might help you design that study. And so we would bring an expert in who's a toxicologist, and we would bring an expert in who's a preclinical planner. And maybe the molecule you'd like to test in that animal model, you only make in tiny little amounts. So that means we might need a chemist whose specialty is in scale-up, how to make grams of that material so we have enough to discuss this. And also how to formulate it. That is, what are you going to mix it with in order to deliver it to the animal to do the study? So we basically have a whole virtual pharma company of know-how that we can bring to each and every project that we're supporting. And that's really what has allowed us to have the privilege of advancing these projects that I've mentioning. So if you are the investigator, then in addition to the money that we provide, our project manager, like Michael Hallen, is going to ensure that we have regular meetings, and you're going to be the beneficiary of some of our planning tools. We can help put together a whole 2, 3, 4, 5-year plan on how to advance until the point where you're ready to go to the FDA. We can bring a regulatory expert to help with that regulatory meeting and the plan and the questions you can ask the FDA. These are the 8 projects that we supported through that original call that I mentioned to you from 2018. And I won't go through all of them, but just to give you an idea of the different kinds of rare diseases they all work on. We had a rare disease like Huntington's disease and ALS, or the Lou Gehrig's disease is what it was originally known as. Also metabolic disease in glycosphingolipidosis, and also really interesting project that we're still actively working with. We've just provided a new grant to this individual in Rett syndrome, which is a genetic disease that affects primarily girls, and where there's a very novel way of turning on the normal gene, which is normally silenced in the girls that have this syndrome. And so as we go through the years of supporting these scholars, and even after they become alumni, we track their success. So here's a timeline of one of the projects with an investigator from UC, the University of Southern California, where we started with just a target, an idea of which genetic abnormality we were going to go after, and then developed a nucleic acid small molecule therapy. And now that small molecule is in a company that the scholar started called AccuraStem, and they're hoping to get into the clinic before this year is over. So that's one of our nice success stories. And then I mentioned the Rett syndrome program. So again, this is a program where we used our experts to help define what are the key experiments that the investigator would need to do. And we're currently mapping the plan to a meeting with the FDA, so we can talk about what the clinical trial design would need to look like. And then a colleague that we have from Louisiana State University in New Orleans. And this is a really interesting program which could affect, or which could be used to treat, if we're successful, multiple different rare diseases that all have a same theme, which is parts of the gene that just keeps expanding and expanding and expanding until the gene that is affected is interfering with many of the neurological and developmental activities of the patient. And this program is under option right now, and we're hoping that it will get developed all the way to a clinical candidate. And finally, I'll just mention that one of the things that we've done in the last couple of years in our rare disease program is to partner with Oxford University in the UK, and together we've launched the Oxford Harrington Rare Disease Centre. Why would we partner with Harrington, or sorry, with Oxford, and why would Oxford partner with Harrington in Cleveland, in the middle of the Midwest of the United States? Well, because we feel that we are complementary in a really in a really exciting way. Our 165 drugs in the making experience we've had so far, and all of the know-how that we have to bring to the table, means that we have a network of really interesting new projects and really interesting investigators. You probably are aware that in rare diseases, we're not going to be able to conduct clinical studies in one single clinic in order to make a program deliver an approved drug. It means we're going to need hospitals and clinics all over the country, and maybe internationally, in order to have enough patients enrolled to see that the drug could work. And so between the University of Oxford, with their 250 different researchers, and our network of 165 plus researchers, and all of their hospitals that they work in, we're putting together that network, and we're also looking for continuing to support projects in the same way that our institute supports projects on the discovery side. So a quick flavor to show you is that in order to help build that pipeline of opportunity that the Oxford Harrington Rare Disease Centre might support, we had a program for applicants to apply from the United Kingdom, and we received 46 applications for our first call, and we funded 5 projects, and those are active now. So we've added those projects to our list, and so it's an exciting time for us. It's hopefully something that gives you some hope to think about, that of the many different diseases that are yet to have a new therapeutic in place to be prescribed. We're trying to make a dent there, and we're incredibly appreciative of everything that makes this work that we do possible. That is the families, the people who participate in the natural history studies, the investigators who treat the patients who we can work with and who have ideas to help us along the way, and the advocacy groups who help to make sure that we have a path forward from a regulatory perspective. So thanks very much for giving me an opportunity to share this with you. And I hope that the rest of your day is a good one. Thank you.
Hello, everyone. I'm Chelsea Judge. I am, yes, Pam Judge's daughter. I'm not quite as a force of nature as she is, but humble to try. Before I close us out, I actually wanted to just provide opportunity for any Q&A. We actually had a couple of questions come in online, so I'm just going to address those first, and then please feel free to ask any questions. I'll either try to address them myself or link you to the right person. So the first question we got was from a patient, and the question is, what can a person do to help further the cause of rare disease, and what kinds of things as patients can we do to get involved? As a patient advocate, sister of someone living with a rare disease, and scientist myself, I would say doing things like this is really great. So being connected with all of the stakeholders involved in rare disease, hearing from Dr. Rensel and learning how you can be a better advocate for yourself and for others, joining with groups like National Organization for Rare Disorders or the Sumaira Foundation if you have that particular condition, right? So aligning with patient advocacy groups, finding your tribe, finding your stripes tribe, I think, so to speak. I mean, there's really a multitude of approaches, and anything you do, whether that's on social media or you're live in person, you're learning about the disease, or approaches for advocacy, I think are all super important. The following question is from a caregiver. And this question is kind of similar, but from a caregiver perspective. So how can caregivers help those with rare diseases, and how can caregivers participate in the rare disease community and help awareness? I'd start by saying similar, like these kinds of organizations, patient advocacy organizations, learning about the disease. Also, I think really learning compassion and empathy and trying to put yourself in that other person's shoes, and then having the humility to realize you're never going to fully understand, but just try, just listen, and be there for your person. Definitely the most important, and anything you do is going to be helpful. If anyone else has anything else to add to either of those questions, please feel free. And if you have any other questions, put your hands up. Okay. By the way, I did not pick that picture of myself. I just want to say that was my mother. She thought I looked like I was multitasking. Well, you know, we all have our different opinions. Anyway. So to close this out, I just want to thank you all so much for showing your stripes in honor of the Rare Disease Day 2023. The theme of this day and just the power of the rare disease community is 100% collaboration. As we've heard the statistics throughout today, rare diseases like those that impact my brother Connor, individually they affect small numbers, but collectively there are over 7,000 known rare diseases. This affects 25 million Americans. This is a public health concern. Rare disorders have a ripple effect. They touch the life of the patient, family, friends, interacting with the healthcare system, government, insurance policies, research, and patient advocates and politicians. Rare diseases are also personal stories of family struggles and triumphs. When Connor became blinded and paralyzed at 22 by this autoimmune disease we heard so much about today, NMOSD, it rocked my family's world. As a sister and immunologist, it was simultaneously terrifying, also fascinating to watch Connor unable to move with these top-notch neurologists poking and prodding him, unsure at the time if he would ever walk again. Out of necessity, my family, like so many others impacted by rare diseases, became obsessed and intertwined with the emerging science and policy decisions that would determine Connor's outcome. Through all that struggle, though, there was hope. There had been rapid interest, progress, and funding into this disease prior to Connor's blindness and paralysis. And for all of those reasons and just the wonderful compassionate care that he received, he did get a swift diagnosis and appropriate acute treatment. And obviously this was crucial. If you meet my brother later today, you'll find that, he has gained his eyesight and ability to walk again. But like many rare diseases, NMOSD is chronic, and that leaves invisible but pernicious symptoms, including permanent nerve damage, for which there are no available treatments as of now. NMO can also come back and with a vengeance at any time, and so to help prevent disease from returning, Connor, like many other rare disease patients, requires monitoring and or treatment to help reduce return of his disease activity. There were no FDA-approved treatments for NMO at the time of Connor's diagnosis. Of course, this is a situation all too common for the rare disease community. But Connor, being my brother and Connor, knew how important it was to support and contribute to ongoing research, and he decided to participate in a clinical trial for a potentially promising NMOSD therapy. Connor, again, is one of many rare disease patients who accepts the uncertainty but hope, not only for himself, but for others, of a clinical trial to help his disease move forward. The Orphan Drug Act, so we're going on an organizational policy level, championed by the National Organizations for Rare Disorders, provided an accelerated pathway and incentive to find and fund treatments and cures for rare diseases. There are now 3 FDA-approved treatments for Connor's condition, and I am beyond grateful to say that with tremendous grit, and again, all of those compassionate clinicians, that Connor is the healthiest that he's ever been since his diagnosis in 2014. So despite all that great work that's been done on behalf of rare diseases, we obviously need more progress, and that's why we're here. We need help to fill in the gaps in awareness, improve patient, caregiver, and healthcare provider education, promote affordable access to comprehensive care, policies that better protect and work for rare disease patients and their families, and of course, more research funding. So I speak to you as a scientist, sister, and advocate, and I learned about how government policies determine healthcare coverage, potential treatment costs, treatment placement on insurance policies, and research funding. And so for all these reasons and more, we need to have a committed ongoing relationship and dialogue with all those impacted by rare disease. So once again, I thank you for showing your stripes and celebrating the rare disease community. Thank you.